in the recording. Today is 11th of September 2025, and welcome to the section Biophysics of Consciousness and DNA Resonance, Biophysics of Consciousness and DNA Resonance at Consciousness 2025 conference. And this is going to be about the various aspects of biophysics and consciousness, and including DNA. So, Irene Caesar, please introduce yourself and tell about yourself. And you can start. You have total 30 minutes plus 10 minutes of the discussion.
First of all, I would like to thank the organizers of the World Congress Theoretical Systems, so Systems Theory, Algebraic Biology and Artificial Intelligence, and the organizer of this section, Dr. Max Myakishev-Rempel, for inviting me to give this speech in Refractional Entanglement for the remote management of biosystems. I am the author of the Fundamental Theory of Wave Optics and Chromosomes, 1985, and the Refraction Code Bioholography, and technology based upon this refractive entanglement in wave optics of chromosomes, and the founder and president of the American company Wave Genome LLC, incorporated in Connecticut in 2010. DNA wave optics provides the theoretical basis for the remote management of biosystems via remote bioholography. It has implications for the national security and is the cornerpoint of the so-called third quantum revolution. Wave Genome LLC develops technology of remote bioholography. And Wave Genome LLC bioholography is based upon the refraction code and refraction entanglement of wave optics in the scalar wave diffraction grating. I had created the fundamental theory of DNA wave optics in 1985, long prior to anybody in the field of bioholography, based upon my unified theory of wave crystals. The unified theory of wave crystals states that the quantum non-locality, zero field, as a singularity, non-local in time and space, is the systematic whole, organized as the wave crystal, that is the tetrapole scalar wave diffraction grating with the paired inverse refraction based upon chirality towards one and only zero center of the zero field, the focus of the quantum non-locality, one and the same for chromosome and galaxy. Information wave crystals is propagated not via some vector signal of vector mathematics or electromagnetic field of quantum physics or regular physics. Vector signal of the electromagnetic nature specifically, but instantaneously and remotely via the refraction towards the focus of the wave crystal of the quantum locality, the zero center of the zero field, with this zero center being one and the same for chromosome and galaxy. The central axiom of my fundamental theory of wave crystal is the axiom of the refractional entanglement instead of quantum entanglement. The axiom of the refractional entanglement is based upon the holographic axiom. The world is 93% energy and only 7% particles. This energy exists in accordance with the holographic axiom, which states that the entire being is contained in its every matrix point. According to the holographic axiom, every matrix point is non-local in time and space and is a systematic whole containing the infinite number of waves, particles, and fields. This is the axiom of non-locality. Thus, every matrix point cannot be defined by any finite value or parameter, such as a particle, wave, and field, or any set of finite values, as well as by such derivatives of finite values as wave matter, the quantity of energy with no mass, or matter wave. The implication of the holographic axiom stated by me in August of 2012 states that the being, if the being is entirely in its every matrix point, then every matrix point is not simply different from any other matrix point, but is unique. This can be called as the axiom of non-local uniqueness of the implication of the holographic axiom. So every matrix point is both non-local and unique. Matrix points differ from each other by their unique refraction codes. And if we have two copies of the same non-local and unique wave matrix, we can transmit information between these two copies remotely and instantaneously, because in the quantum non-locality, where there is no space and time, they constitute one and the same systematic whole. Thus, the entanglement between two copies of the same non-local and unique wave matrix is not the quantum entanglement, but the refractional entanglement. Here comes the axiom of the zero signal. The fractional entanglement is the zero signal, or the holographic signal, non-local in time and space. The zero signal cannot be defined by the principle of locality in time and space. The non-locality of the zero signal implies that every matrix point is both uncreated and indestructible. Thus, no artificial neurons or artificial neural links, which are created and thus finite, can be in principle defined as matrix points capable of zero signal. The same is true of any artificial computational chromosome, or any artificial DNA, or any artificial microorganism or organism. Science based upon notions incapable of accessing the zero field is doomed for failure. Thus, we should define the matrix point and the zero signal and create the zero signal technology based only upon the wave optics and refractional entanglement theory. The matrix point axiom. The matrix point is the systematic whole capable of A, procreation of oneself as a whole, that is the female gender chromosome, B, capable of being conscious of the infinity and all its implications. The matrix point is capable of procreation of oneself as a whole, precisely because it is capable of being conscious of the infinity and its implications. Conditions for the matrix point. Scalar wave diffraction grating is created by a coherent signal with permanent frequency, which is long enough to be scaled to get the scalar wave diffraction grating created from macro to micro dimensions and vice versa ad infinitum, thus creating refraction. Annulment axiom of the zero field. Each segment has its own resonance frequency, which is annulled by the scalar wave when the peak of the forward-going wave is nullified by the trough of the same wave when this wave is reflected back upon itself. Again, the holographic or zero signal cannot be described simply as any electromagnetic signal or the limited structuralist theory of isometry, such as fractal theory, which cannot account for the uniqueness of each fractal in the systematic whole. The chirality axiom. The linear isometry of the fractal theory cannot also account for the tetrapole inversion of the signal by the scalar wave. The inversion of the signal by the scalar wave is defined by the chirality axiom. I repeat, in the wave crystals, including chromosomes, information is transmitted not by some finite linear signals with certain electromagnetic parameters, but remotely and instantaneously, with no limits in time and space, via refraction towards the focus of the wave crystal, with the focus zero center being one and the same for chromosome and galaxy. That is, information is transmitted via focus of the quantum non-locality as the wave crystal, the zero center of the zero field. The chirality of the zero field axiom. The quantum non-locality zero field is the ultimate wave crystal with the two paired concave lenses inverted in relation to each other. Clear vision and regeneration are possible only when brain is in the theta brain wave state, which is a scalar wave, with every segment of the scalar wave diffraction grating focusing man's unique refraction code down to a chromosome. The same gene gets expressed in the functional individuals and species via metacentric chromosome, which is like equilateral cross and analogous to the well-centered and focused eye crystal. And the same gene gets expressed in the dysfunctional individuals and species via acrocentric chromosome, which is half of the metacentric chromosome, and is analogous to the far-sighted or nearsighted eye out of focus. The same gene in apes is expressed by the acrocentric chromosome, and in humans by the metacentric chromosome. The same gene is expressed by acrocentric chromosome in the wild horses, which are like donkeys, and by metacentric chromosome in the domesticated horse, which is superior to the wild horse by its physical and cognitive abilities. Children with Down syndrome differ from normal children by the presence of extra acrocentric chromosome. Male gender chromosome is acrocentric. That is degenerative and dysfunctional. That is why males are incapable of giving birth. And female gender chromosome is metacentric, that is functional and regenerative. That's why females are capable of giving birth. Chromosomes function as lenses, literally changing their shape like our eye crystal, and so their aperture of their scalar wave diffraction grating to receive information remotely and instantaneously via the quantum non-locality zero field. The structural damage in chromosomes is the major cause of the disease. Thus, the neural and genetic engineering should not be allowed to go along the path of inflicting the structural damage to the systematic whole of the matrix point. Since every matrix point has its own unique refraction code, including a functional chromosome, the future of the 4P medicine, personalized, preventive, predictive, and participatory, belongs to the bioholography based on lasers, which are the only devices producing a coherent signal needed for creating the scalar wave diffraction grating. Using various lasers, Wave Genome LLC develops and produces the wide array of the generators of the form for optimizing the wave optics of chromosomes in the analog and digital form. Thank you very much for your attention. Questions are welcome.
Irina, questions? Anybody?
You start, Max.
I say, I guess my main question is something about quantum sensors and artificial intelligence connected to consciousness and DNA. Specifically, what practical applications do you see around, especially commercial products? What should we pay attention to?
Well, my company is a very successful business enterprise, and before the war, military special operation, the monthly profits were in digits. So, and my company is not simply does research and development; it does production of devices that enhance the wave optics of the brain wave activity, the wave optics of chromosomes in the brain, which works with the refractional entanglement for the digital bioholographic pharmacies. The company that produces devices both in digital and analog form as generators of the form for creating the scalar wave diffraction grating with specific application of unique client's unique refraction code. So every client has his own or her own refraction code. So to apply the so-called 4P, preventive, personalized, predictive, and etc. medicine, participatory medicine, we need to work with clients specific, unique refraction codes. So we need to develop or to have as a company, a commercial company, we need to have devices that are capable of reading and recording clients' individual, unique refraction codes. It is possible only via lasers. Our in the 21st century paradigm, our ancestors, they used the analog form using the operators, so some specific person, right? Because humans are, humans are like lasers, and there are humans that are even better than lasers. So our ancestors used operators who were fasting to avoid introduction of superfluous homologous nucleotides to the chromosome, so that these superfluous homologous nucleotides will not destroy the structure of chromosome, because chromosome is literally a lens. So it's a lens with this refraction code. And so they were not eating meat, pork was forbidden, casual sex was forbidden, sex for procreation was limited by ritual and procreation itself. Incest was forbidden, homosexuality was forbidden. So all processes that were introducing superficial extra nucleotides to the chromosome, which is supposed to have a precise number of nucleotides, no more and no less, and in a specific geometric configuration. So this introduction of superfluous homologous nucleotides was limited. These people were called saints. In Russian, it's святой, the person who emits light. So the person who is capable of focusing light. So this person was fasting. He was or she was avoiding these activities. Also, this person was applying electrodes, which are generators of the form which produce the scalar waves. And these electrodes are polymer crystals, precisely polymer crystals of the animal origin, such as fat from bears, geese, and other animals, of insect origin, such as propolis, from minerals, such as shilajit, mumio, from plants, and all Ayurveda, all natural medicine, folk medicine is based upon electrodes. And they were eating electrodes, drinking electrodes, applying electrodes on their body. So here comes the ritual of mummification. So these people were mummifying themselves during the lifetime. So you cannot become a high priest without all these conditions. So become an operator to record and transmit the refraction code of the refractional entanglement and controlled by systems and other people remotely, instantaneously. So you should go through all these rituals. and they were applying electrodes in the liquid crystal form. That's how we got this notions of God anointed king, or to anoint for the kingdom. The king was anointed with the frankincense. Then our ancestors applied electrodes in the aerosol crystal form. That's how we burn the incense in the churches, burn the frankincense, and then the solid crystal form. Every Russian man or woman was wearing ladanka. Ladan is a frankincense, is a pendant with the solid resin polymer crystal of frankincense inside this ladanka. Besides this, they were praying. Prayer was the ritual of putting your consciousness into the theta brainwave state. Rosary, when a Muslim is doing his or her rosary and saying Allahu Akbar, or Christian who is doing his rosary and saying God is Christ is King, and etc. The prayer. This is simply the way of putting one's brain into the theta brainwave state. These people also were living in the dwellings, houses, which were the generators of the form of the scalar wave. They were living in the cupola ceiling rooms, houses. And these houses had domes, the bells, they were ringing the bells. And, Nepalese Buddhist monks, they are putting this, I'll show you, they are putting this huge singing bowls on their head. And like this, you put it on your head, and do like this. So with this simple procedure, analog procedure, you can get out of the very serious medical conditions almost within 5 minutes. It's like a miracle. It's proven by experience. So they were living the life which had the morals, social code based upon scalar wave. Do not do upon others the things you do not want to be done upon yourselves. So it's the whole system. So you can, so our ancestors, they used humans, specific humans, who were putting themselves into the state of being the generator of the form for producing the scalar wave diffraction grating.
So Irene, can you
have technical devices? So we have
Irene, can you tell us what
produce this technology?
Irene, can you tell us what frequency the bowl should be?
This is the whole point. It is not about frequency. The initial frequency to initiate the scalar wave diffraction grating should correspond with its wavelength to the size of the resonator to get reflected back upon itself. This is the whole thing. But our ancestors, they created the generators of the form in such a way that this, I don't have the bell here. So this generator has different diameters. You see, each diameter corresponds
different frequencies.
To its frequency, to a specific initiating frequency.
Can you give us your website, the actual
But
name?
anyways, the golden rule is we have 5 senses, right? So if you hear the sound, this already means that you are capable of inversing the sound to create a scalar wave. This is the golden rule. So you should not go after the frequency. You should go after the generator of the form. So you create your analog generator of the form or digital generator of the form, but the digital should have again all the characteristics of being the generator of the form. And these characteristics consist of 2 major parts. First, you have the resonator, which is capable of inverting this initial signal, reflecting it back upon itself. But again, as I told you, if you hear it, you already are capable of doing it, right? And then the second part of the generator of the form is that you should have a conductor or semiconductor that doesn't allow the scalar wave to get extinguished, because it gets extinguished. It's a crystal. You know, we use sugar and salt to preserve vegetables, fish, meat. Why? Because crystals, they produce scalar waves, and scalar waves annul the waveforms of viruses and bacteria, you see. But if you consume too much crystals, it's not good for you. And they also, scalar waves, they extinguish themselves. So you should always have the conductor and semiconductor as a part of your generator of the form for the scalar wave diffraction grating. And my website is wavegenome.com.
What is your website? Can you give it to us?
Wavegenome.com.
Say it again.
Wave
Glenn,
Genome.
it's in chat already.
Wave Genome.
Yeah.
Okay.
Yes.
It's in chat. If you open chat, then you'll see it.
Oh, okay. .com. Okay.
Any more questions?
Yes, may I ask?
Yeah, please go ahead.
It is very interesting, but of course it needs a lot of additional study because very complicated. My question is about chirality. You were talking about chirality. What? So as we understand, that's optical purity, L and D, and so on. Do you mean this meaning of chirality, or you were talking about some other?
Chirality is the structural parameter. You see, my theory, the structuralist theory, better described in the classical terms as Aristotelian basically theory. It's a structuralist theory. So in chirality is a structural term. We have vectors, we have so-called scalars, right? And chirality is a mirror, it's a mirror parameter, is a parameter of a mirror, inversive reflection. It's a reflection that inverses the geometrical optics, you see.
I see. I see. Because when you begin to speak about chirality, I, as biologist, begin to think about the very sophisticated problem of optical purity in biology. There is no, in all living systems, have very optically pure amino acids and carbohydrates. They first are only L, others are only D. And this is the indication of a living state, well, which was discovered by Pasteur. The major question, what is the origin of this optical purity? Because it is 900% optical purity in living matter of biomolecules. So what is the origin? Because in non-living in matter, there is no such such purity at all. When we die, our molecules begin to racemize, racemic, yes. L comes into D, D into L, and so on. Does your theory can it explain this, for example, or suggest something about the origination of this optical purity? It has to do with to all molecules, and also not only on the monomer level, but when we are talking about DNA maybe it is spiral, and it has also optical purity, and higher levels of organization also have optical purity. Did you think about this in your theory?
Well, I would like to offer you, make an offer to write a paper together on this optical purity.
Okay.
No,
Thank
no.
you very much.
uh,
Yeah,
the- You
I
can
like.
take email from Max and connect with me, and we can. I'm regularly publishing in this Восточная Сибирская академия, в открытой академии.
Okay.
Каждые 3 месяца публикую. Давайте его там спокойно опубликуем. And to answer your
Надо
question,
подумать.
of course, the whole point of my speech was to prove that human is a measure of everything. So it's this anthropocentric view of the ancients was completely correct. You see, and the divine nature of man and divine rights of man, they're all scientific notions. You see, and the closer we are to the zero center, the more of the optical purity we get.
Okay, I agree.
Thank you, Irina. Thank you, Vladimir.
May I ask a question also?
We are running out of time, but go ahead.
Just 2 simple questions. Hi, Irina.
Oh.
We have been talking. I'm Anna.
Yeah,
We
I
didn't meet, but yes, with Glenn. It was wonderful presentation. I admire what you're doing, and just I'm your big fan. But my question is about clinical trials, which probably have been done, if you tell just briefly, on a living system such a multidimensional cells like and one layer cells and in vivo and in vitro. It's number 1, and number 2, I'm very curious about matrix city, and would you please tell us?
Well, I have produced 1 clinical trials because it's complicated, very expensive, but I have produced it on a very high level with the Bauman State Technical University, which is the largest and most important university in Russia of MIT level, using lasers, which are at that point newly invented lasers, and the mathematical paradigm for this experiment was produced by the same scientist who produced the mathematical paradigm of the trajectories for the new Russian middle range missile caliber. And the experiments, the clinical trials were for aspirin, holographic aspirin. And so the biohologram of the aspirin was recorded by the laser and administered via digital holographic aspirin, and as the generator of the form, electrode-based chip applied to the applicants 24/7 with successful results. And these successful results were published recently in 2025 by the peer-reviewed scientific journal.
Wow. Oh, can I, maybe I'll just email you, because I would like, because I read your articles, but I couldn't find, some answers which I had in my head. Yeah. It would be nice to try it on living organism and cells, especially, yeah. And about matrix.
But I'm very open to any kind of, joint mutual experiments on the American soil, because the Wave Genome is an American company, and I'm not going to transfer it anywhere else from the United States. And in regards Matrix City, it was the Matrix City Consortium with the Institute for National Security in Moscow. And these were the people who actually employed Garyaev in the end of 1990s. So these were those people who had this aura section within the huge научно-производственное объединение in Russian, scientific research enterprise Energia with 5,000 Soviet scientists. And these people conducted the remote transfer of insulin much prior to Garyaev for the American Diabetic Association. They administered digital insulin 20 kilometers over 20 kilometers to the diabetic calf successfully, and so on and so on. So this Magic City was the consortium, Magic City, and from that time on, the only thing that I did for this concept, I built the house with a huge cupola, 7-meter diameters, and 3.5 meters high, perfect hemisphere. And this is, and so basically I built the huge generator the form.
Unbelievable. I wish I could come there.
Welcome. Yeah, it is by me too. So, yeah.
So it's kind of quantum teleportation or something like that. Yeah.
Right.
It just like sounds wow, bravo. Thank you.
Thank you for your kind words.
Max, you should
Max.
switch on your microphone.
Oh
You're
yeah,
on silent.
thanks Irina, thank you very much, and thank you everybody. And Irina's website is in the chat, wavegenome.com, and you can find Irina's contacts on the website dnaresonance.com with email. Email is there. So next speaker is me. Thank you everybody for coming. I have Anna, Glenn, Vladimir, Sergey, Irina, Victor, Andrew, Howard, and Mark. I should remind you that, just a sec, I will mute microphones. I'll remind you that you can turn on automatic captions. Вы можете включить автоматический перевод. The button is in the Zoom on the bottom. It's in the frame. It's CC in the frame. So in the right part of the toolbar, and you can switch spoken language and the language you want to see. So the captions is there, and it's a additional feature in the Zoom.
May I ask a technical question?
Yes, go ahead.
Please, will this transcript recorded? Can we get it?
After.
The video I'm recording, yes. The transcript, YouTube automatically transcribes again, so you can see transcript there. Yep.
Thank you.
I might transcribe everything and automatically and send it out. I will see if it works.
Transcribe it and put it in a book.
Yeah, I mean, I really thank everybody for coming. It's a wonderful. I see familiar faces and familiar names, which I know are interested in DNA, resonance, and consciousness. Okay, now I invite a new next speaker, which is me, and I will start my presentation. I'm Max Myakishev-Rempel, and I will speak about biophysics of consciousness and DNA resonance. Can you see my screen?
Yes.
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 So I should highlight just a sec, I need to move that away. I should highlight the website dnaresonance.org, and you might know it from because you came here through it possibly. So this talk is about Freire model, and I published the whole. There's a chapter which is self-published. It's not published yet. It was invited to a book, but it's not published yet. So if you go to DNA Resonance website, you click on, so video presentations will be here, full text publications, click on the full text and you get to that. And this Freire model is on the next page. It's called Rempel Consciousness Freire 4M. It's a big chapter, which I will try to squeeze here. The whole problem of connecting consciousness and DNA is huge and complicated. It's not trivial. We don't know a lot, and a lot of things are transdimensional. So we really have very few ways to reach there. But there are paths of connecting back and forth, and I will show a few connections. I will start from the model that we published a while ago, which connects DNA DNA in the nucleus. So DNA is in chromosomes in the nucleus of the cell. And Khamerov and Pentros suggested that microtubules are important. And I think the argument is very strong that microtubules are transmitting and participating actively in the work of cells, and especially in the work of neurons transmitting information. So that's Khamerov, and he's basing that idea on the action of anesthetic drugs and on geometry of microtubules and on the function of microtubules. So what we suggested is that DNA vibrationally is coupled to microtubules. Microtubules are in cytoplasm, and they come close to the nucleus, and actually they connect to the nucleoli, and they plug into nucleus vibrationally this way. That's a hypothesis, not proven, but if you look at the photographs of microtubules, then perfect candidates for actually transmitting the information. And the next idea is that DNA in one cell, that's the problem, how DNA can talk between the cells vibrationally. So DNA in the nucleus of one cell is connected to the DNA in the nucleus of another cell through microtubules. They vibrationally connect. They go to connection between neurons, which this is axon, and the connection is called synapse. So in synapse, they don't touch each other, but they come, if you look at the microphotographs, they come very close to each other. So there is a gap. And this gap is called synaptic junction or synaptic gap. It's a major, well-studied gap, synaptic gap, which is regulated by pretty much all psychologically, psychically active substances. So this is well studied, and all the famous dopamine and serotonin and GABA and other transmission and receptors are regulated through drugs in this gap. So I learned this idea from Richard Alan Miller that resonance property of this gap probably defines a lot of qualities of the mind and psychology and emotions. So it's not only chemistry, possibly it's a resonance of the gap is the key to consciousness. So that's a simple model. That the DNA of the body is connected into one unified vibrational system. I will just go through highlights. The next topic is what could vibrate in DNA, and here we pay attention to the genome. So genomic sequence is known. It's 3 billion base pairs, and if you each chromosome is about 10 centimeters. If you stretch all the chromosomes in one line, it's about 2 meters. It's all packed in a tiny nucleus, which is about size of 5-7 micrometers. And if you look at the genomic pieces or genomic sequences, the well-studied part of the genome is called protein coding exons, protein coding part of the genes. They're called exons. And its total protein coding part of the genome is 4.5. So that's what is studied, well studied. The rest is called junk DNA or non-coding part of the genome. And we believe that this is the part which is actually takes part in resonance and consciousness, the non-coding 95.5%. And from this part, introns are also part of the genes, and they're not translated into proteins, but they're part of the genes, the insertions in the genes. And repetitive part of that is 24%. Why are we talking about repetitive? Because for resonance, you need to have multiple units to vibrate and resonate together. So repetitive part is what we think is actually responsible for resonance and structure and structural properties of chromosomes. And the rest of the genome, which is 46%, is intergenic sequences. And of them, repetitive is 28% of that. So total together, it's over about 55% of the genome is repetitive. So that's huge, and that's, I think, the key to consciousness and to the function of resonance function of the DNA. Jump into the next topic is the proof that DNA is actually genetically important. We know that children are similar to parents. Even if they were separated at birth, they still inherit the properties of parents. So we believe DNA is important. The mainstream science believe that DNA is super important, but Sheldrake, Rupert Sheldrake, questioned the importance of DNA, and he demonstrated that actual estimates of contribution of DNA to the phenotype, the heritability, is hugely inflated, and I agree with him. So the most of the studies that prove that DNA is important, they use twin studies. So they do twin heritability estimates, and this, in my estimate, in my table, describe the details are described in the chapter, but basically they are pretty much double or even more, maybe they triple the actual genetic heritability. So true genetic heritability is less than a half. The rest of variance is something else. We can talk about that, but we are not only genes. There is other factors which contribute to the phenotype variance. Okay, so inflation in twins, I believe, is happening because twins synchronize in their womb. So they develop together and they become synchronous in the womb, and that's why they are more similar to each other than the fraternal. The identical twins are more similar than fraternal twins. So genetically identical twins are more similar because their DNA synchronizes. So that's why the twin study overestimate genetic heritability. So it's called missing heritability gap. It's well known, and it's kind of swept under the carpet by geneticists because it helps them to get more funding. If genetics defines, say, 80% of, say, in schizophrenia, twin studies show 80%, but if you actually measure in GWAS studies, it's much less. It's less than 40%. Usually it's 10 to 30% heritability. So, but DNA is still important, just not that huge. It's less than half, maybe a third of the variance. But still, in that how do you call it? In this true heritability, it is resonance, I think, contributes a lot. So the non-coding part of DNA contributes a lot, and proof for that is that if you look at the non-coding part of the genome and conservation in the evolution, it is it is also well conserved. There is, between the genes, there are conserved sequences that show that DNA is important. That non-coding DNA is important. Okay. Next, I jump to the next idea is nature of consciousness. And here I just accept the model which is known for forever. It is in all cultures that there is a universal consciousness, and that we that personal consciousness is part of the universal consciousness, and that the mind, so that's the human body, that the human body is part of the universal consciousness, and that it cannot be separated, that the illusion of separation is is just illusionary. And in this picture, I'm trying to place the DNA in it. And in the body, I place DNA outside to show that this, in my hypothesis, is that DNA is the actual interface between the mind and universal consciousness. And much of the mind exists outside of the body and is connected to our physical conscious mind through DNA. So basically, through resonance and other information transfer, the universal, the external subconscious mind is connected to the internal mind. So that is part of the Frere model. And the second part basically says that the outside world, this green outside world, is created by as a simulation, is created from inside out. It's not only my model. I just accept the well known idea popularized in the movie Matrix that this is a simulation. I don't say it's computer simulation, but it is natural simulation in which the outside circumstances are created from inside out. Proving it is pretty much impossible because that simulation, it seems to have a rule that it protects the illusion of separateness and protects itself from being discovered. So from inside out, you cannot really prove that you live in simulation. You can have a personal experience, but sharing with others is prohibited. And there is a lot of protective programs that prohibit us from proving to others that we live in simulation. So the key from this slide is that physical body has a skin, and the skin separates us from outside. And it is an illusion that mind is located in the body and that mind is located in the brain. And this illusion is created by through resonances in DNA. Okay. Then there is a dilemma which is pretty much also unsolvable. I just kind of plotted on the picture that we know that consciousness creates. No, the claim of this theory is that consciousness creates the body and creates the brain. So consciousness creates the brain, and brain creates consciousness. And that dilemma is solved by dividing to universal consciousness and individual consciousness. So the brain develops through experiences individual consciousness. But that is all part of the universal consciousness shown on the previous picture. The next idea is we need to solve this riddle to explain the true role of DNA in consciousness. So it's unavoidable. The next idea is to understand the flow of time, the illusion of flow of time, and the free will. And here I demonstrate, it's called bump and go car. And you all know them. They were from the 50s and 60s. These are the cars which go, and when they bump into an obstacle, they rotate the wheels inside, and then they choose a new direction as if they were self-driving cars. So that existed even before electricity. So that's one of the examples of self-driving behavior. So to make choices, you can use this mechanical or largely mechanical model, which makes similarly choices. So what is free will choice? It's the choice which is made in the absence of full information and under the pull of time, under the pressure of time. if you had unlimited time, you probably would choose the perfect solution. If you had unlimited time and unlimited information, you probably could calculate the perfect solution for any problem. But because of the pool of time and limited information, you have to make so-called free choice. So there's a free choice and pool of time are critical. And again, I published a paper where we suggest that DNA plays huge role in creating the illusion of pool of time, illusion of time. The next idea is idea of fractal rendering. So rendering, you probably know from computer games, where in a computer, I don't know if you play computer games, but at least you watch people playing computer games. And in many games, they are multiplayer games, so multiple individual consciousness are in the game. And you walk through the world or fly through the world, drive, and as you walk through the world is created in front of you. So that is idea of rendering, and it's also pretty well known. The idea is that the reality doesn't exist until you pay attention to it. So as you walk through the world is rendered as if it was stable. But that idea of rendering is huge. And the next idea of fractality is that you are a fractal part of the universal consciousness. You're a fractal fragment of universal consciousness. So the idea of fractality that I'm coming back here, that the main trick to understanding the role of biology and DNA is to understand that the illusion of body, illusion of mind, and illusion of time is created by separating us from the rest of the world. And what's inside the skin is a fractal mirror of the outside world. So what basically it's a mirroring, it's transformation, mirror transformation, fractal mirror transformation. What's inside out, what's inside is a mirror of what's outside. So that idea is, I guess, is it has many names, but one of them is monism, another one is solipsism, and it is known for a long time. Here I just wanted to combine to bring four main players. DNA sequence contributes to mind through many ways, and the main claim of this presentation that DNA dance is how that happens. So DNA dances and produces thinking. And then also DNA contributes to morphogenesis, the shape of the body. And also it produces, through the shape of the body, it produces the brain and the mind and the personal individual consciousness. Okay, next I need to suggest that it's impossible to understand the role of DNA and in consciousness without looking at glitches in the matrix or glitches in a materialistic model of view. And one of the key breakthroughs, I don't know if you're familiar with that, happened just recently, a year ago. Actually, it's like 9 months ago. Telepathy tapes came out, and they are led by Kai Dickens. And now they are created a big movement pretty much all around the world, because it was discovered that telepathy is real, and it's measurable, and there are very good telepaths around. So the main discovery was that autistic, non-speaking, non-verbal children and adults are usually telepathic. And when the normal people, they're called I'm blanking on the name, neurodivergent versus neural something. When normal people start teach non-speaking autistic children typing, then this telepathy is pretty easy to measure and prove. So telepathy tapes is a documentation of that community growing, discovering telepathy in non-speaking autistic population. And this population finally got a voice. They publish books, they make videos, and in many cities now there are communities where people finally discover the telepathy. And if you know Strugatsky's prediction in book Гадкие лебеди, I don't know the translation, bad swans, ugly swans. The idea that the humanity evolves into the new species. So the idea is that this new autistic population is the next branch of human evolution. They're genetically different and they possess telepathy, and they create a global telepathic network. And that is the future of humanity. Moreover, they can teach normal people to be telepathic, and they do that. So now their teachers and caregivers are telepathic, and they talk to each other, and they even teach lessons in silence using telepathy. I did my research on telepathy since 2009, so I have sufficient experience to believe that it is true from my personal research. Carl Jung is a pioneer in psychic research and also in telepathy. And in 1933 letter, he said that the existence of telepathy in time and space is still denied only by positive ignoramuses. And especially revealing our experiments with telepathy in dreams, dream telepathy, and psychic clear vision in information transfer in dreams. The key researchers here are Diane H. Powell, which is the main academic researcher who stands behind the telepathy tapes, and her book ESP Enigma, the scientific case for psychic phenomena, is one of the key books about telepathy and psychic phenomena, and she explains how brain anatomy contributes in psychic phenomena. Dean Radin wrote Real Magic and so many other good books, and he is one of the main researchers, and he summarized big paranormal research, parapsychology research, especially in telepathy and information transfer. And Rupert Sheldrake, Morphic Resonance, and other books are on that topic. So these are three main names in academic alternative science about psychic and telepathy research. So from that, we basically understand that there is a pretty good body of evidence that actually the materialistic worldview is incomplete, and the only solution to solve the consciousness problem is through paranormal research and actual quantum physics. Here I just highlight the ideas that DNA contributes to body in two ways. It goes through producing RNA, and then it goes through producing proteins, and then proteins make up the body. So from physical matter to the body, DNA creates the body, and changes in DNA sequence create changes in the body. And also there is a second idea proposed by many researchers, including our collaborator Richard Miller, that DNA produces biofield and interfaces with biofield, and biofield also programs the blueprint of the body, the shape of the body. And then we start looking more closely at DNA, we realize it is not naked, it's not pure double helix, it is wrapped around nucleosomes, and that also, so this is like pretty well-studied model of a nucleosome. This is a pretty well-studied model of tetranucleosome. And these are very orderly structures. The DNA is very orderly. Nucleosome is a little bit less orderly, but pretty orderly, pretty regular. And tetranucleosome is the largest orderly structure of DNA. It's still pretty small. It's in the order of under 20 nanometers. It's pretty small. But after that, DNA is wrapped into chromosomes in a similarly disorderly nature, in similarly disorderly way. So there is no known larger orderly structures of DNA. And that's basically, after that, it becomes a soup, which is disorderly. But so we're trying to find the logic in that chaos of DNA. And it seems to be the key to understanding that life is based on combination and dance between order and chaos. So also in understanding this picture, it's important to realize that DNA is hydrated, and it's very essential to understand that water structure is tightly linked with the structure of DNA, and it's pretty much impossible to decipher the role of DNA resonance in the body and biofield in the body without understanding the structure of the water. And we'll have speakers today that speak about that. So water structure, like we all know snowflakes, but the mystery here, how that part of the snowflake is synchronized with that part of the snowflake. And that, I think it's still a mystery. I don't think anybody asked that question and addressed that question. Somehow they are coordinated, and there is no known mechanism that would explain that coordination, because local conditions here and local conditions here are different. So, but there is a coordination and synchronization between 6 branches of the snowflake. And that's another mystery to be addressed. Next, we started looking at water structure around DNA. And I stumbled upon the idea of Gerald Pollack that a flat water structure, which he calls easy water, the DNA imprints on these flat structures, imprints its own structure, helical structure. So I started looking and discovered that this flat structure was first described by Lippincott in 1969, and it's based on discovery of deragging of polywater about 10 years before that. So there are several names, including Deraggin. Actually, there was someone before Deraggin. I'm sorry, I don't have in front of me, but in the chapter, I spell out this history. It's possible to find the history how polywater was discovered, and then it was sort of discarded by mainstream science, and it went into being one of the prohibited, discarded parts of the science, as usual. So what is important about this polywater is it's in Lippincott that it is not exactly perfect. It has gaps and canyons, and these canyons allow it to be electrically neutral. If you fill the gaps with structure, it will become negatively charged. So there is a dance which is continuous of building and dissolving that structure. So that dance is super important. That is a slide from my 2023 talk where I say that DNA from one cell talks to another cell through waves, and these are nucleosomes, and basically the structure of DNA is important to transmit the wave information, and then it can go from structure to waves, and then from waves back to proteins to structure. Next slide is that nucleoplasm is very thick. There is liquid water, there is chunks of crystal water, liquid crystal water are DNA, and it's 1% DNA, about 20% of protein, and the rest is water. So it's very thick and it's constantly moving. And this movement is actually shown on the video. It's well known. It's called chromatin reorganization. It's a sped up video, but it's still very fast. And it is a whole nucleus. That part you already seen before. And I see that someone is already sleeping. Hey. All right, so here I looked at I tried to decipher the idea of Pollock that layers of polywater are shifting, and I realized that these shifts can imprint the sequence of DNA can be imprinted on the layers. So I created that model where layers of polywater are actually perpendicular to DNA, and they're shifting as in pin tumbler lock. So as pin tumbler lock has a key classical key has peaks and basins. The pins in the pin tumbler lock go up and down, and the same idea is that as DNA changes its shape dependent on double helix and the sequence, the layers shift right and left. And in the first layer, it is a stepwise movement in polywater. It can move in six positions, and the next step it can move to another total. I think if you count these dots, it's after 2 steps it can move into 18 or 19 positions, I think. And that's just another illustration of shifts in polywater, how they reflect the DNA sequence, not only double helix, but actual sequence. That's a nucleosome, and showing that parallel DNAs probably are bound by structured layers of polywater. And the idea that polywater is built around negative structures is also belongs to Pollock. He studied that well. So that if you put like charged surface, then polywater is built around this surface. Here I expand the idea of disagreement between the structure of DNA and structure of water. The polywater is hexagonal, has hexagonal symmetry. DNA has hexagons, and it has a bit of hexagonal symmetry right here. No, right here. Hexagonal symmetry in DNA, so these are hexagons. But if you look at double helix, right here, its symmetry actually is 10.5 terms, 10.5 steps per turn of the double helix, and in two turns it's 21 steps, 7 multiplied by 3. So in 2 turns, 1 turn and another turn, it's 21 steps. So there's 21 steps per 2 turns and 6 steps per turn. They disagree between each other, and that, I believe, makes DNA uncomfortable in water. It's on one hand, it's hydrophilic, and it is bound to water, and it exchanges protons with water. On the other hand, I think it vibrates back and forth, spinning back and forth, trying to fit in water structure, and that, I think, is the basis of life. It's the main basis of energy, and that's why we are warm-blooded, to get to produce our 36.6 degrees to increase the vibration of DNA in water. And because DNA is charged, that creates oscillating electromagnetic fields in water. So the whole dance in the nucleus is a dance which is acoustic, electric, and electromagnetic with with lots of frequencies. So there's, I think, is one of the keys to DNA resonance. That is just another illustration of how DNA symmetry is disagreeing and conflicting with water symmetry. Next, I jumping forward. I'm close to the end. Jumping forward. That's a larger DNA structure, and I propose that repetitive sequences. There are multiple families of repetitive sequences called transposons. They are not positioned in DNA in order. It's a semen disorganization. They are pretty random in DNA. I suggested that positions of these repetitive elements called transposons in the DNA sequence create a bigger structures of DNA by coupling them together. I called it homological coupling. So it doesn't really matter the idea is that it doesn't really matter what repetitive sequence it is, as long as you have two similar sequences in the DNA strand, they will attract to each other. And there is some quantum theories suggesting that similar structures, similar sequences of DNA would attract in water and couple. So it's a weak coupling. It's a new force. It's not well described, but the idea is of couplings creating the new structures. And because of Brownian movement, Brownian motion in water, it's a live dance of forming and dissolving structures, larger structures of DNA. And next I went to test this using genetics data, known genomics, published genomics data. And the method is called Chromatin conformation assay. It's published. There is a lot of those, many of those, not a lot, but I would say hundreds of data sets published, good quality. What they do is they freeze the structure by adding formaldehyde and linking all the proteins together. So this structure is frozen in time. They take cell culture, add formaldehyde, freeze the current structure of DNA. Then they cut it with a restriction enzyme, or it's called micrococcal nuclease enzyme to cut. Then they ligate using ligase, and that creates combined ligation junctions and sequences that combine together. So they were far away from each other, but due to ligation, they are now captured together, and that is sequenced using standard inexpensive sequencing. Once you do that, you know what stood in a cell, in live cell, what was near each other. And that's allows you to test whether, test that hypothesis that repetitive sequences come together. So hooray, I found that they do come together. I learned with, first it was done with a hired programmer, but since artificial intelligence became good in programming and inexpensive, I learned how to program myself using AI. So it's pure vibe programming. I speak in English and it does the programming. I run it on my own server in Amazon instance, in Amazon virtual machine on Linux. And for the last 4 months, I was doing that. Finally, the results have been submitted, hopefully in today or tomorrow. What you see is that different transposons called MIR-B, L2B, 3N, L2C, 3N. They are enriched near the ligation point where things were ligated. And moreover, they are specific. So similar transposons come together. So basically that idea is verified. I cannot prove it's causal. It could be just a coincidence that in pieces that come together, these transposons are specifically coupling together, but now I have a metric which actually demonstrates that. And that's homotypic specificity. Homotypic means the same family, say MIR-2B, is coming together in two strands. So if there are two pieces of DNA, if they are linked, if they're contacting each other, the chances that they occur together are elevated near that junction, near the contact point, and they fade away to background level far away. So I think that's I'm pretty confident that's real. And it's reproducible in biological replicates, and it's reproducible between different individuals. So the coming together or going away from similar. So these transposons that like to come together, these are transposons that push against each other and actually repel each other when they're together. They don't like to be in contacts. And this idea of not liking being together, I think, is also very, very essential. So in many cases, the signal comes from the sequences repelling each other specifically. So the L2C 3' transposon repels and prevents the DNA from contacting. So I think these are two factors going together. This is a slide going through all my presentations. There is a lot of frequencies starting from infrasound, which is fractions of hertz, to very high frequency, which is X-rays and ultraviolet. And these all frequencies which are important. I ran out of time, but I should highlight the idea that quantum entanglement in biology is super important. It's pioneered by Philip Ball and explained that much of health and biological signaling goes through quantum entanglement, and that quantum entanglement is spread through radiation and Brownian motion. And the reason sun is healthy and is therapeutic for us is because the rays of sun are coherent, and that coherence is absorbed by us, by eyes and skin, and makes us coherent and healthy. So coherence is given to us through quantum entanglement, through light. Same thing with healthy food. Coherence is the key, as many other speakers would probably will mention, that coherence and quantum entanglement are key to health and also to consciousness. The last couple slides I will show that we are forming global telepathic network. It's already happening. It will happen through natural ways, through normal people learning telepathy, and through artificial ways, through using telepathic helmets. It's called synthetic telepathy, and that's also coming. Currently, it's done through Neuralink, which is surgical, but there are already helmets which are optical, and they also work through quantum entanglement. So it's possible to actually connect to technology and become telepathic electronically. Check out Richard Alan Miller's paper from around 1980, 1990 about synthetic telepathy. So that happens, and another key change for join us in one telepathic network is artificial intelligence, which also will help uniting us into a more coherent system. It's a bumpy road, but I think it is inevitable. And I will end with the idea that it's super important to understand ecology of DNA vibrations, of DNA resonance, that the whole biosphere is one united vibrational system, and that this ecology is strongly affected by electromagnetic means and also by genetic manipulation of plants, animals, and humans. So the genetic manipulation of plants and animals is fully in progress, and there is a lot of genetically modified organisms. And genetic manipulation of humans is illegal in many countries, but there are countries where it's still not outlawed, and it's so easy to do that it's very likely it's already happening. And it's mentioned by some of the military leaders that genetic creation of soldiers is also attractive to them. So genetic manipulation of humans right now happens without understanding the vibrational ecology, and that will damage the collective vibration of humanity. So discovering of the DNA resonance and vibrational ecology is essential for survival of the species. And again, I highlight the site dnaresonance.org. You can find the whole chapter there. And again, this is a Frere model suggesting that we are a mirror of the universe, fractal mirror of the universe. And I thank my collaborators, Ivan Savelyev, Oksana Poleskaya, Michael Rempel, Ekaterina Erenpreza, Nelly Zarianova, and Richard Alan Miller. And these are my past workers. my collaborators, Irina Konstantinova and Vadim Gushin, who already are crossed over. And this is my contact. Thank you very much. I think I used all my time. I guess I'll take one question if you have.
May I have a question here?
All right.
Yeah. Thank you very much, Max, for your very lucid and very interesting presentation. I would like to emphasize that you have a fractal rendering or fractal mirroring from internal mind to external world. Although it is also known yet there is a mirror brain that or mirror neuron that work such in such way, but I think we could look to other field. For example, Stephen Lehar, if you already know, Stephen Lehar work about this Gestalt theory of space that he hypothesized that our brain actually create the space by consciousness.
I agree with
Maybe
it.
it's, yeah, maybe it sounds more like a back layer. There is no reality, but the space itself is created by the brain. But I think it is interconnected. Yeah, like you say that the consciousness affect the brain, then the brain affect the consciousness. And the second, I appreciate that you mentioned that we should try to discourage people, especially researchers, to modify human DNA without concern of vibrational ecology and consciousness. Yeah, and what I'd like to ask is one, how the interaction between consciousness and brain, that people may call it neuroplasticity, can be used to help, for example, in the case of dementia or Alzheimer, yeah, because many people are growing old, and maybe there is a way to train again the brain to rejuvenate. Yeah. I had a relative that have a case of dementia and Alzheimer.
I got it. Thank
Yeah.
you.
And
It's a
the
big
second,
question.
oh yeah, okay, yeah.
I'm running out of time. I'm over time for 20 minutes, so I need to invite next speaker. But I wanted to highlight the idea of coherence, and that coherence is key to not only neuroplasticity, not only to shape of the neurons, but to inner health of neurons, and which is linked to health of chromosomes. And this is chromosomes in neurons, the chromatin in neurons and chromatin in the rest of the cells. It's damaged by environment, but also by the mind. It's both are important. So coherence in the environment is important, of the food and vibrations, but also coherence in the mind is super important. So yoga, I just discovered that yoga is one of the great tools for bringing coherence, and the meditation in yoga and many other Tai Chi, Qigong, Reiki, and other cultures, other natural healing techniques are greatly help coherence in the mind and in the body, and I believe it will help the chromatin, but also technological introduction of coherence is also important. Finally, after the red light therapy was discovered in 1968, many years ago, but finally about 10 years ago, there was green light from FDA in the United States. So now a lot of red light therapy devices are finally on the market. And I think it also works through quantum entanglement and bringing coherence to the body. So technologically, it's also possible to increase coherence in the body, not only chemically, but through waves. And I will stop here because my time is over. And I would invite the next speaker.
Thank you very much, Max. Yeah.
Sure, thank you very much. I thank everybody for joining. And Glen Rein, you can turn on your screen sharing. And it's we met with Glen at Tesla Tech conference several years ago, but maybe 5 or over years ago, and maybe 7 years ago. And Glen is actually the one who does experiments. I do computation mostly, and Glen is actually not only Mr. DNA resonance in theory, but also in experiments. So Glen, please, you have your 40 minutes, where 30 minutes is intended for the talk, and I will just slide a little bit behind, and 10 minutes for discussion.
Thank you,
You're
Max,
good.
for the introduction. Can everybody see me and hear me?
You're
See
good.
the slides?
The visual is good, and the sound is good.
Okay, you can see the slide. Okay, so there's the title of my talk.
You can close the format background by pressing the cross, so it will be bigger, or you can press F5 to make it full screen.
Yeah, I'm not sure exactly how to.
On the bottom left, there is like a little screen on a tripod. You can click on that, or F5 button. F5.
Oh, that didn't work.
F5.
Yeah, F5. Okay, try
on the
F5.
keyboard. F5.
Okay, so
Try again. On the keyboard, F5.
I'm looking for F5. I don't see F anything.
Oh, you don't have it?
That's strange. Yeah, this computer doesn't have F
Okay,
anything.
go to the bottom of the screen. The right there is a slider, and next thing, bottom of the screen. Bottom to the right. There is a slider, and there is a button there. Right to the right. Even more right. Four icons to the right. Even more. This one. Yep.
Slideshow. There it is. Okay, great.
Yep.
Okay, everybody, thank you for Max, for the invitation to talk about my latest research. Yeah, I'm an experimentalist, so I'm going to talk about some actual experiments. And what I'm going to propose in this particular model, here's the title, which you can read, about an alternative to scalar energy as the new buzzword, and a new kind of energy to consider, and that is imaginary energy fields. Because imaginary fields are described in the physics literature, and they've actually been measured. So it's a whole new way of looking at energy and how this kind of energy is involved with particularly DNA. So, in my previous slide, previous talks, I've talked a lot about quantum energy and quantum energy fields, and have developed this sequential model to describe the relationship between different kinds of energies, starting with the most subtle energy on the far left, which you could call it ether, or you could call it divine energy, and that the sequence of how these energies are related is described here. That etheric energy gets converted to virtual energy, or Okay, so now I want to use the word imaginary, but we have virtual energy and quantum energy, and then the quantum energy underlies, controls, and regulates electromagnetic energy, which in turn affects the physical body. And this last arrow on the far right is well studied in the bioelectromagnetics literature. But the question for me then becomes, is it possible for electromagnetic fields and or the mind to actually regulate and control this flow of information? Because this is ultimately how the body gets healed with energy. And if we can do that with our mind, then we need to understand this relationship. And for this particular talk, instead of using the word biology, we're just going to substitute DNA. Okay, so then the question is about imaginary numbers and imaginary fields. And I just, as a way of introduction, want to talk about complex numbers, because complex numbers have both a real and an imaginary component. And complex numbers are the fundamental of quantum physics, where the imaginary numbers are used in the equations, and the equations don't really work without the imaginary numbers. And in the scientific literature, believe it or not, I've seen the term magical properties describing the nature of imaginary numbers. So, and in addition, they talk about complex numbers preceding real numbers, meaning that they're more fundamental, and they are associated with imaginary spatial dimensions. Well, this is exactly what we're after when we talk about consciousness and the relationship between our physical bodies, or our DNA, and consciousness, because it's the higher dimensional nature that is most likely to describe consciousness and its interaction with our bodies, and that imaginary numbers are an integral part of the spatial higher dimensional realities. Okay, so there's a little bit of, that's not even theory, it's right out of the physics literature. Okay, so then the question for me becomes, well, what is the relationship between virtual energy fields, imaginary fields, and quantum fields? Because the question for me is, are these three independent types of energy, or are they three terms used to describe the same fundamental energy? So one way to look at that is to look at the scientific literature, which I do, and it turns out that these energies are related, at least in this case. Virtual energy and imaginary energy is described in this particular article, where they talk about virtual photons in imaginary time. This is a published article. Sorry, I don't have the actual reference, but you could look it up technically with the title and the author. Okay, so, and here we have another article describing the relationship between imaginary and quantum, looking at the quantum mechanical understanding of imaginary time. So therefore, these things seem to be related. But these are, the mathematical concepts. And I want to now look at some of the physical terminology and parameters that are used in the physics literature. All this stuff comes right out of the scientific literature. So we're going to start with the idea of imaginary surface tension, because this particular article talks about imaginary surface tension and actually measures imaginary surface tension. And in the next slide, we can see the data that, this is another study by Flatter in 1999, where they actually measured water that had a little bit of iron in it, and measured the imaginary surface tension, and you can see, and the real, and you can see on the far left the line which shows the relationship between real and real surface tension and frequency, and on the right the graph showing imaginary surface tension versus frequency, and you can see that the imaginary surface tension gives much more information than the real surface tension. So this is a good start about why we want to look at imaginary numbers and not real numbers, or I should say, in addition to real numbers. Okay, so now let's look at imaginary impedance, which is another parameter that is measured and described in the physics literature. And in this case, it's related to phase information. And in this particular article by Sapuan recently used the imaginary impedance to calculate the impedance. Okay, they used impedance tomography imaging, which is based on imaginary impedance. And here you can see the real comparison. The real imaging, the real tomography, is on the left, and the imaginary impedance tomography is on the right. So in this case, it's not a question of more information, it's a question of different information that's obtained when you use the imaginary numbers. So another good reason for looking at imaginary numbers. So here's a graph that came actually out of my computer, showing the difference between real and imaginary impedance as a function of frequency. This is up to 100 kilohertz. So in the high kilohertz regions, you can see that there's very different information in the imaginary impedance compared to the real impedance on the top graph. Okay, so that was my introduction. And now I want to talk about the original research that I've done in my lab using impedance spectroscopy, technically electrochemical impedance spectroscopy, which involves a potentiostat and two electrodes, which you can immerse in a solution of water or water containing DNA. And I've done both. And this particular technique is standard, but I've modified it a bit, to say the least. Okay, one of the things, the particular instrument that I use comes from Gamry in Pennsylvania, and this allows me to adjust the voltages and the frequencies of a particular measurement. So I use very weak voltages, around 30 millivolts. I use a very unconventional form of electrodes, which are not cylindrical wires, but the wires wrapped into flat Tesla coils, which are counter-wound so that the two electrodes are wound in opposite directions. The two electrodes are very close to each other, and they're made of dissimilar metals, particularly silver and gold. Well, the use of silver and gold is quite interesting because there's here 3 different articles talk about the exotic properties that you get when you use dissimilar metals, noble metals, as electrodes, where you can actually generate a Casimir force in between these two electrodes, and this setup allows for electrical tunneling effects. So the modifications that I've made to the standard way of measuring impedance is significant and allows for me to have a very sensitive method of measuring very subtle effects on water and DNA. So the question then is, what is the application for this particular new method? And the first 3 methods I've actually done myself in my lab, and they include the measuring of water and DNA in water, imprinted with different energies and different frequencies. When I say different energies, I mean different types of energy, which you'll see in a minute. I've also used this, in essence, to study the electrical properties of biomolecules like DNA, and also recently to measure cells, living cells from my body. They're called buccal cells in the scientific literature, but really they're cheek cells, which you can easily obtain by taking a knife, scraping the inside of your mouth, and transferring these cells to water. I'm not going to talk about that research or the water research in this particular talk. I'm only talking about the research with DNA, as Vladimir says, in water. Okay, but the method can also be used in principle. These two electrodes could be put on an acupuncture point, and it could be put on plants. I haven't done these kind of experiments, but just in case there's any experimental researchers in the audience, here's a couple of ideas. Okay, so now let's talk about the actual protocol that I use to take a measurement. So to start off with, the first thing I do is use my Clever scope, which is an oscilloscope combined with the spectrum analyzers, to obtain the frequency signature of a particular sample. In this case, we're talking about DNA, and we're talking about the fact that there are frequencies that can be measured in the between 1 and 100 kilohertz. Then I would choose the strongest signal, and which I would call a resonant frequency, and use that particular frequency to take my impedance measurements. So again, my impedance spectrophotometer allows me to adjust the frequency and to take measurements at a specific frequency, in this case, a resonant frequency of the target system. Commercial spectrophotometers and impedance analyzers don't allow you to do that, and you're stuck with taking a measurement at some arbitrary frequency like 2 kilohertz, which is, not ideal to get the kind of sensitivity that we want. So I actually record both the real and imaginary impedance before and after treatment of the water with various forms of energy. The measurements are in kiloohms, so it's like an impedance resistance measurement, which obviously this audience knows is reciprocal to conductivity. So in essence, I'm really measuring the conductivity of DNA. So I take the sample before and after treatment. I take 3 aliquots and then take the average. And this experiment done in the absence of any treatment with sequential measurements gives the experimental error of about 5%. And in this particular talk, the data that I'm going to present, the raw data that I'm going to show you, is presented as a percent change relative to the untreated control or the before value, before any kind of treatment. Okay, so that's the protocol. And DNA, that's what we're going to talk about. Oh, I use human DNA for the record from Sigma. It's double-stranded fragments of placenta DNA obtained from female placenta. So they pool it and they extract the DNA and purify it, and it's 99% pure DNA. And then I, suspend it, make a solution in water, and stick the electrodes into the solution. Okay, there we have it. Okay, so what we're in essence measuring is the electrical properties of DNA, and I guess for this audience, this slide is probably redundant, but just to remind everybody that DNA it can either be a conductor, a semiconductor, or a superconductor, and all of these different states are described in the physics literature. Basically, we're talking about a charge transfer of electrons and protons, which can either move down the central channel of the DNA or on the outside surface. And as a result, the DNA exhibits quantum tunneling behavior. So there's a little bit of an introduction. I guess most people know, but just in case there are no scientists in this group, that's why we're studying the electrical properties. Okay, so here's some experiments that I did, old experiments that I did, just in case other people don't know my research, with the real impedance measurements when exposed to a healer. I get similar results when exposed to electromagnetic fields. But the point I'm making here is that on some days, the effect is to show an increase. And in this case, we're talking about, again, impedance or resistance, but conductivity is the opposite. So in some days I get an increase, in some days I get a decrease. In other words, the treatment is higher than the control or lower than the control. And this is kind of assumedly due to the environment and the fact that there are all kinds of energies in my lab, electromagnetic energies, all kinds of energies. So this is kind of annoying because if I'm treating the sample with a specific kind of energy, like the energy from a healer or an electromagnetic field of a specific frequency, I have a lot of interference with the energies of the environment until I do the experiment inside a Faraday cage. And then you can see the same experiment done on sequential days, and I'm getting the same, different magnitude of the effect, but the same direction. So yeah, some days, on day 4 here, it was a very weak effect. And on day 2, I had a very strong effect. Well, that's something I can live with. I can take an average, and in this case, that horizontal line is the average. And although there's a large standard deviation, at least I have a number that's consistent. Okay, so let's talk about electromagnetic fields and scalar fields, since we heard a lot about scalar energy in Irene's talk. This particular method of generating electromagnetic fields and scalar fields was developed by Bob Beck, who believed that if you take a coil and put a spot weld in the center of the bar where the wires are wrapped around, that you generate a scalar wave. So I had my engineer, Al Kaspersky, build a setup just like shown here, with and without the spot weld. So I can generate an electromagnetic field and a scalar field of exactly the same parameters, which are sort of outlined here, with the difference being that the two coils are out of phase in the scalar mode and in phase in the conventional electromagnetic mode. And the fields that, a little more detail here are, quite strong, 100 Gauss and 50 ohms. And I'm using strong fields because I want to be able to measure a real practical effect on the DNA, physical effect on the DNA. So let's start here again on the where we're looking at real impedance on the y-axis. And in most cases, the impedance goes down and the conductivity goes up. But at different frequencies, and it can either go up or down. 42.8 kilohertz is the Keeley frequency, where he discovered that frequency actually implodes water. But for most of the studies I've done, I've used 528 and 432 hertz because they're solfeggio frequencies, and they're important for the body and for healing purposes. Okay, so the results of these kind of experiments that where their direct comparisons show that electromagnetic and scalar energy have similar effects on DNA, but that the scalar is significantly stronger. So DNA resonates better with scalar energy than with a regular electromagnetic energy of the same frequency, 432 hertz. Okay, so moving on, the same experiment was done so this was done with a sine wave, and here we can see that the sine wave gives the same kind of effect. Here it's like a 35% effect, and here it's a 45% effect. It's really a large effect. But when you do it with a square wave, you get an even larger effect. Imagine that. You have all these harmonics present. That's not surprising. But again, this is all with real numbers, real impedance. So now I want to move on to kind of the etheric properties of DNA, because I actually did an experiment with measuring not electrical properties of DNA, but the etheric properties of DNA with a radionics instrument, as depicted in the bottom right. I did this in collaboration with one of my colleagues, Peter Moscow, in the U.S. Psychotronics Association organization. And what we did was we stimulated the DNA with a plasma device, causing it to unwind, and then measured the rewinding, the spontaneous rewinding of the two strands every 30 seconds with both a spectrophotometer and with the radionics device. And these two devices were separated by 1,500 miles. So we were just curious to see if there, if there was any correlation between the two measurements. The spectrophotometer measurements were done at 260 nanometers, which is a classic way of measuring the hydrogen bond connection between the two strands of the DNA. The more unwound the DNA, the more accessible that wavelength of light is, and the stronger the signal. So what you get is over time, as the DNA rewinds, you get a decrease in that signal, and here we have a comparison between the radionics measurements and the spectrophotometer measurements, and you can see that there's a peak at around 90 seconds after in both the radionics, the etheric energy, and the physical energy of the DNA. But what's most interesting is that around 50 seconds, you get a peak with the radionics device, which is absent in the spectrophotometer, suggesting that there's some kind of etheric energy change in the DNA that precedes the physical change that can be measured by the spectrophotometer. Okay, there's the conclusion from that interesting study. Okay, so now let's move on to measurements of impedance in response to different types of energy. And here I'm showing you the raw data, the before and after, the percent change, and the ratio, which is obtained by taking the ratio between of the percent change values. So most of the time, that ratio, like you can see in the electromagnetic treatment, is around 1 or less than 1. In fact, 90% of the experiments that I've done the ratio is less than 1. And the imaginary number ratio, sorry, it's the imaginary divided by the real number. That's the ratio when I say ratio. So the higher that number, the stronger the imaginary component is to the measurement. So it turns out that scalar energy has a ratio of 1.8, which is significantly higher. And a healer, this one particular healer, has a ratio of 1.7. And various healers obviously have different ratios, and in a way, perhaps it's a measure of the effectiveness of a healer. Can't say that yet, but it's certainly a measure of their etheric energy. It seems. Okay, so here's some other examples of types of of treatments, energy treatments, and their corresponding ratios, which in this case are all way over 1.7 and 1.8. This particular healer on the top was an Indian healer who claimed that he was generating nadi energy that for the first time on this planet, and that and his numbers show a rather high ratio of 2.8. The Therify is a plasma device which has counter-rotating plasma fields generated by two separate flasks containing inert gases, which are rotated in a counter-rotating manner. And then you put the sample of DNA in the middle, and that gave a ratio of 2.2. The next two experiments were done with sound waves. The solfeggio frequencies were generated with a tuning fork, and they did all 9 solfeggio frequencies and recorded it. And I played the recording of that sound to DNA and got a ratio of 3.5. So that says a lot about the energy generated either from a tuning fork or from a specific frequencies of solfeggio numbers, all of them. And the last example I'm giving you here is a recording of a chant, if you will, chanting the names of God in Hebrew. That's why I'm calling it the divine names. And this was done by J.J. Hertak, and the recording shows a rather relatively large ratio of 1.8. I put it in a loop so that I could expose the DNA for about 60 minutes to this kind of energy. The DNA, responds to virtually all energies I've ever tested, but it's a slow responder. These effects take an hour. You don't see, except with healers, of course, which can be a lot less time. But the DNA needs time to absorb and resonate with the energies in its environment. It doesn't, and to respond, to change its conformation, to change its electrical properties, whatever is going on physically that we're measuring here at the flow of electrons, etc. So that's the data that I have, and I'd like to conclude my study by saying a little bit about imaginary fields, which have been measured. It was a recent publication where they actually measured a quantum spin system, and they measured the quantum coherence in a spin bath. And they're actually called Li Yang zeros, which is what happens when the partition coefficient goes to 0. And it gets really technical really fast, but it's published in a mainstream journal. First time, as far as I'm aware, any kind of subtle energy has been measured. So imaginary fields, therefore, are something really to consider, and something to consider as an alternative to scalar energy as being very important in the body. Anyway, the point is that imaginary impedance can be measured with a spectrophotometer. Imaginary impedance gives different information than real impedance, and that I've used the imaginary impedance here in this study to characterize the effects of electromagnetic fields. That's my time's up. And human DNA to characterize the effects of healers and other forms of energy on human DNA. And it is worth noticing that in some cases, imaginary impedance shows effects which are not measurable by real impedance. So it's more sensitive. And that the ratio of imaginary to real impedance is the most sensitive. And therefore, I want to conclude and introduce to this group a new parameter, a new experimental way of measuring and quantifying the effects of subtle energy on DNA. Thank you so much, everybody, for your attention.
Glenn, thank you very much. And invite questions. There is a lot of people. Can you hear me?
Yeah, I can hear you now.
Yeah. I invite questions. Any questions?
Glenn, I have a question.
Yes,
You
sir.
mentioned the properties of DNA, electrical properties of DNA as conductor, superconductor, or insulator.
Yeah,
Did
right.
I understand you correctly?
Yes. Yeah, that's from the scientific literature. That's not my research.
I see.
Apparently, I mean, when I give a non-technical talk,
I see.
most people didn't even know
Thank
that
you. Yes, thank you.
DNA
Because
conducts electricity.
yes, In a few minutes, I'll talk about another example of superconductance in living matter. Yeah,
Right.
and I have another question, which the question which I would like to clarify for myself. Can you just, in few words, explain difference between imaginary impedance and real impedance?
Well, so I have a
Basing on
spectrophotometer.
some analogies, for example, you see, because, well, this is a very sophisticated physics, but maybe there are some analogies which will clarify a little.
Well, so the simple answer to that is, from what I've read, and actually, there's a whole book called Imaginary Physics that I'm working my way through. So, the imaginary impedance is associated with, if not a measure of phase information, and unlike real,
Uh-huh.
which, right? So that's the best I can answer that so far with my knowledge. I'm obsessed with this concept of imaginary impedance, as you're pointing out. It's a new idea, but it's in the scientific literature. There's
I see.
several articles that actually show the real and imaginary components. And I'm not a physicist, Vladimir. This is a
Yeah.
better question for you and a physicist
I'm
in the
not a
audience.
physicist, also I'm a biologist.
Oh, that's right. Oh, I'm sorry. Oh, right. You are. I forgot. Yeah, right. So we got to ask some physicists. But apparently the ratio of imaginary to real is something that electrical engineers use when characterizing circuits. I don't know if that really helps, but and they claim it's a measure of the phase properties of the circuit,
Uh,
therefore of the DNA.
I see. So, can we say that imaginary impedance has something to do with alternative currents, while real with direct currents?
No, I don't think it's
If
that
we are
simple.
talking about, if you use the word phase, so when you use the word phase, then we have something alternating, yes,
Yeah,
and
yeah.
yeah.
Right, yeah. And the spectrophotometer that I use uses it's all AC stuff. So DC impedance may or may not have an imaginary component, but AC impedance certainly does. And my
Mm-hmm.
spectrophotometer, it
Yeah.
takes a measurement and then it calculates the real and the imaginary components. And you would think
Mm-hmm.
that they're mathematically related because they come from the same measurement. Then the machine actually calculates it too. And in 99% of the cases, the real and imaginary components go in the same direction. You,
Mm-hmm.
you know, the if you before versus after a given treatment, but every so often I see that is not the case where they go in opposite directions. So
Mm-hmm.
I didn't show you that data because I don't know how to take a ratio. I mean, I guess you could take the absolute value of the changes that occur, but
Mm-hmm.
they're not in the same direction. So it's another whole uh, observation about
Yeah.
imaginary numbers
Mm-hmm.
and the effects of subtle energy on DNA.
Yeah.
I
Glenn.
just
Mhm.
Googled using ChatGPT version 5, and it says imaginary impedance tells you about the energy storage and relaxation processes in the solution, while the real part tells you about the energy loss. Essentially, it's resistance versus storage.
Oh, yeah. I did read that. I forgot about that. Right.
Mhm. And
Right.
can it have something to do with coherence
Oh,
and non-coherence, like imaginary impedance? Again, when we are talking
yeah. That's
phase
a
and
good
so
question.
on. Yeah.
I don't know the answer
Yes.
to that.
That may be because if you have to do something with coherence, it's very different from
Yeah.
the classical our understand. There's no coherence in classical our notions, and here
Right.
Yeah,
That's
and imaginary
right.
then it may be coherence, and it may have its specific length, its specific time, and things like this.
Well,
Which
okay, so I can actually answer that question with some experiments that I did with water,
yeah,
and when if we all agree, especially you, that that structured water is more coherent,
yes,
then it
that's
turns
what
out
I
that
mean. Yeah,
structured
that's what I mean.
water actually has a lower ratio than regular water by a factor
Hmm.
of tenfold. I didn't include that data, but there's a huge difference in three different types of structured water. They had a much lower ratio,
Hmm.
whatever
Okay.
that means.
Mhm. Thank you.
Yeah, the ChatGPT agreed with you. Essentially, coherence in water means more structure, and with more
Hmm.
structure, the energy storage.
Yeah, exactly. So it seems there's a reciprocal relationship between the coherence and impedance, or at least
Hmm.
the ratio.
Well, at least they're relevant to each
Yeah,
other.
yeah,
Again,
yeah.
my
They're
question is,
related. They are related. Yes.
Related, yeah,
Right.
related, yes, but
yeah,
relevant
my
at the
question
same
is, how can we produce therapeutic or other practical applications based on what you learned? Or diagnostic or therapeutic.
Well, yeah, well, that's the impedance tomography is used by the medical establishment. At least, well, I don't know. That's a question for a doctor, or a radiologist or something. It's different information is the point. And we need to do a whole bunch more experimental work to allow us to understand exactly to answer that question because we don't know at this point.
Yeah, I just want to comment that I worked on the same floor where there was a device which used a bioimpedance for measuring cell culture properties like growth and other
Yeah.
and health. So bioimpedance is now
Oh, yeah.
a commercial product essentially. It's pretty
Right.
expensive, but it's like a dollar, $100 per 96 well plate just for consumables. It has golden electrodes printed on a 5 millimeter well. So you get 96 wells. You put culture there, and through that electrodes you measure impedance. It's not
Well,
as fancy as your gold and silver, but it's at least it is it's
it's
real.
close,
It's mainstream now. It's
and
sartorius.
and we need to just have those people, those practitioners, looking at the imaginary part of the impedance. Apparently,
If you can do that, they should be able to do it.
yeah. Well,
They
right.
just need some imagination.
So, but you can buy the impedance spectrophotometer that I have for $9,000, so it's affordable.
Right. Yeah, I looked at the pricing. Yeah, the one which was suggested to me was 25,000, but I know they're basically. I'm very interested in very high frequencies like
Oh,
gigahertz.
yeah. Oh, well, yeah. If you want to go to, yeah. Right.
But gigahertz is above 25. It's more
Yeah,
like
but remember, the body functions at very low frequencies.
it's all over the place.
Yeah.
But gigahertz is millimeter waves,
Yeah.
42 gigahertz,
Yeah.
and I have that device. The device is pretty cheap. It's Milta. It's called Milta. It's a Russian made. And when you,
Send me the information.
yeah, I will. It's a
I would love to
therapeutic
work with
device.
gigahertz
Yep, it's a
fields.
therapeutic device. It has this three gigahertz frequencies. One of the like 56, 42, and 37 gigahertz.
Oh, 42 is
And
great for water.
and when you put that generator on your skin, the mood changes like in seconds.
You feel it.
You
Wow.
feel it, the
Wow.
mood, and the pain gradually goes away. I overused it, and then I get some health crisis, so I don't actively promote it because it's so powerful it can hurt.
Wow.
Well, it took some weeks for me to recover. It was just kind of
Really?
shock,
Wow.
energetic shock. But yeah. So frequencies are cool. I'm sorry for dragging the time, but I wanted to bring it to practical applications.
Yeah, great. Thank you.
Now, I think, Glenn, let's talk not only in my webinars, but in between webinars, we should speak more. And now I invite the next speaker, Vladimir Voeikov. Please, Vladimir is well published in mainstream as well, in English writing journals, and is expert in biology and water. Thanks, Vladimir.
Okay,
for
thank
joining.
you. Yes, I'll try now.
We can see, you can do the, perfect.
Yeah. Do you see it now?
Yeah, see and hear clearly. Thank you.
See and hear. Okay. First, of course, I'm very thankful to Max for invitation to this very stimulating and informative session. And already from the previous talks, I got a lot of new ideas for me. And I think that probably thanks to Max, we will have opportunity to discuss them later on in his webinars. I believe. Yes, Max. I think.
Yeah, thank you for wishing that, because I was waiting for people to ask to do more. And if you want, of course, we will do more.
Yeah. Okay. So. First, I would say that I'm not a professional in consciousness. Nevertheless, I think that I know a little about water, and it came to me that without water, we cannot speak about consciousness. Of course, we can speak, it is difficult to speak about consciousness without DNA, but unfortunately, I don't have enough time to discuss the role of DNA in consciousness in relation to water. And thanks to Max, we already have some ideas. He talked about this a little. Now, when we use the word consciousness, the problem is that we are talking about a very vague problem. And George Miller, who was the founder of the cognitive science, here's a citation for him, that consciousness is a word worn smooth by a million of tongues. And depending upon the figure of speech chosen, it is state of being, or we may use a word soul. It may be a substrate or some kind of a field. It may be a process, because consciousness cannot be static. And it may be an emergent, as he says, aspect of matter, in particular of brain. So it is a very vague notion, but nevertheless, almost everyone agrees that consciousness is, at least in part, a property of our brain. I suppose that not many people will argue with this claim. In fact, when we are talking about the consciousness as a property of our brain, then we may start to study it as a property of brain, because now we know a lot about, and the physiologists, neurophysiologists talk a lot about altered states of consciousness, and comparison of normal state of consciousness and altered states of consciousness may help to understand what normal consciousness, or in general what consciousness in fact means. So there are now very strong methods to study state of brain and changes in the state of brain. and even to get not only qualitative but quantitative parameters of the state of consciousness. One of these methods, very sophisticated and very widely used in medical practice, is a method which is called functional magnetic resonance imaging. And recently, I refer to a paper, to the pioneer paper, which was published in 2016 in PNAS. They were measuring function. I'm sorry, just I'll switch off. They were measuring the state of brain using functional magnetic resonance in normal state of a person or after LSD application. And what was found? It was found that under LSD, as compared to placebo, the visual cortex increases its communications with other areas of the brain. So this functional magnetic resonance imaging gives us a lot of information of the state of the dynamic state of the brain of a living person whose state of consciousness change from so-called normal to so-called altered state of consciousness. In fact, profound effects of LSD in brain has been visualized. I will not talk about the results. It is a very interesting paper for not only for neurobiologists, but for psychologists, psychiatrists, and all the people who are interested in different states of consciousness. But I am now pay your attention to the method. Because this functional magnetic resonance imaging allows to show the state of brain. In fact, this method directly evaluates the state of water in cells and tissues. And that's a great pity that most medical doctors or most professionals in this field of fMRI completely forgot how this method just originated. It was invented by the American engineer, Dr. Raymond Damadian, who was basing on Gilbert Ling's idea of water structuring in living cells. Gilbert Ling is just the founder of this whole direction of studies of water in living cells. And Raymond Damadian, who knew the principle of nuclear magnetic resonance, which measures the state of spins in particular atoms. In particular case, it measures the state of spins in protons, and it is called proton magnetic resonance. So this proton magnetic resonance can be used to study the state of water in a sample which we analyze. So, basing on these Gilbert Ling's ideas, Damadian decided to compare state of water in normal and cancer cells. I will not go into details of the method itself. Many people use NMR, nuclear magnetic resonance, and they know how it works. But in principle, this method allows to see the state of protons in this or that wet sample. In particular, in the whole living, for example, in mice in general, or in part of its tissue. So when we study the state of protons, and people at least know that they studied the state of protons, most of us forget that more than 99% of all protons in any living matter belongs to water. So, in principle, when we study the state of protons in any wet sample, we study the state of water in this sample. And water may be more dynamic, more chaotic, or more structured, more organized. So what Damadian has shown, and he published this paper in this 1971 in Science. Unfortunately, he was completely forgotten, and this paper was also forgotten, which was entitled Tumor Detection by Nuclear Magnetic Resonance. He has shown that water is more constrained in normal tissue, and it gives dark color on the picture. If you represent the result like some image of a tissue, and cancer signal shows that water is more chaotic. So in normal tissue, there is a lot of constrained water, and in cancer tissue, there is much less constraint in water. But not only in normal and cancer tissue, the state of water changes. So these different states of consciousness, this is placebo, this is LSD, they show that the different changes or states of consciousness is different states of water in different parts of the brain. After LSD, brain is much more united. Water is much less constrained to particular parts of the brain. Here is the result of water changes in brain after visual signal. So after visual signal in normal state, only visual parts of brain, water becomes less constrained. After LSD, the brain behaves as a unit, as just the whole unit. So this method already shows that state of brain and state of consciousness, because we are talking here about normal and altered state of consciousness, are closely related to state of water in brain. There is another example which demonstrates important role of water in consciousness, which follows from the quantum theory of consciousness. Marx already told about the works of Hameroff and Penrose, and I will not talk a lot about microtubulars. Thanks to Marx, I can skip a little this part of my talk. But what they were talking about? They were talking about that anesthetics, including, for example, inert gases like xenon, which cannot chemically somehow modify any receptors and so on, these anesthetics, they irreversibly modify function of microtubules, which are abundant in pyramidal neurons in cortex, by blocking their coherent terahertz vibrations. These microtubules, they coherently vibrate, and these vibrations are responsible for the characteristic feature of consciousness, awareness. So Stuart Hameroff, being an anesthesiologist, he just noticed, and or at least claimed that after anesthesia with xenon, not the only function in the body which disappears is consciousness. The human being loses his consciousness. When xenon leaves the body, the person recovers completely and becomes again gets the normal state of consciousness. Together with Penrose, they developed quantum theory of consciousness, which suggested that entanglement, coherence, and quantum computing occur in brain microtubules, offering potential solutions to challenges in cognitive neuroscience. So this theory is 30 years old already, but during all its history, it was under very severe critics from from medical doctors, from biologists, and also from physicists, who said that there cannot be any quantum physical effects in such warm and wet substance as brain matter. Nevertheless, in last years, during the last decade, there appeared a lot of confirmations of this theory, that there may be some quantum physical effects related to the state and changes of state of microtubules. But one of the strongest supports in favor of their theory was the recent discovery of a role of water in microtubulus functioning. It was discovered in 2013 that microtubules having such a channel inside it contain highly structured water inside and probably outside of them water, which hydrates the outer surface of a microtubule. So this water, as it has been shown, is capable of resonating in terahertz frequencies, and it may be even some type of quantum photonic laser. What was also shown that this interstitial water channel outside microtubule is solely responsible for the 100,000 times more conductivity than tubulin. Now there are claims that even this water has the property of superconductivity. And that's why there was my question to Glen about superconductivity properties of DNA. It was demonstrated that dry microtubule is an insulator. There is no any electrical conductive properties of tubulin polymers. This is dry microtubules. But when water is constrained in it, water becomes a superconductive substance. So this water core should act as these authors who published this paper, and it was supported. There are now about 300 references to this paper of Sahu, of the paper of 2013 years. So this water may act as a current source by storing charge. So that's why it may be superconductive. That means and superconductivity, as everybody knows, is the solely quantum effect. It's not it cannot be explained in the realm of classical physics. So there were two examples of water functionality in the in object or subject which we call the cradle of consciousness. Now let's talk about water in more details. I call water not a substance, not a matter, but I call water not only substance, not only matter, of course, but water is its nature. It is a structured process. What it means? First, before what it means. Now, Philip Ball, one of the persons who studied water a lot, talked about water a lot, consultant at Nature magazine, and he was 20 years ago very conservative, talking that there is nothing special about water. Now he writes that no one really understands water. It is embarrassing to admit it, but the stuff that covers 2/3 of our planet is still a mystery. Still, nevertheless, we know now more about water than about consciousness. And we already have seen that consciousness and water may be closely related to each other. And so if water and consciousness are interlinked and we penetrate deeper into water science, our understanding of what consciousness is may also deepen. So let's try to penetrate deeper into water science, about the novelties in water science. But first, we'll start from the ancient times. And the founder of the ancient Greek philosophy, the founder of philosophy, as we may say, he claimed that water is the principle of all things, that all things are created from water and all things return to water. So it was said 200, 2,500 years ago. Now, just very recently, science proved that water not just is really the principle of all things, because water is the second most abundant molecule in the universe after hydrogen. So the Herschel discovered, oh, I'm sorry, discovered abundant quantity of water in which contains about 150 10 power 12 of the oceans on Earth, in places where the new planets are found. So such planets are around quasars, for example. So this it turns out, and if I don't have time to tell you or to remind you that water was found recently on Mars, even on Mercury, on Venus, on Neptune, and so on. There are satellites of Saturn and Jupiter which consist on 60% of liquid water. That is very important. So in any case, first, water is second most abundant molecule in the universe. So it is known since only for 14 years. This is a very important, I would say, claim. So then, what we know already about water, that water is not just a passive matter, though it is considered like this by many biologists and physicists. It is an active process which is capable of self-organization. And water molecules are capable to form dynamic ensembles with each other. Dimensions of ensembles vary from cosmic, like on this previous slide. This is the water vortex, cosmic water vortex, containing tremendous quantity of water vapor, of course. From cosmic to macroscopic vortices in flowing water, to clouds, to tornadoes, and to nanoscopic dynamic structures, already quantum structures, which are called coherent domains. This is the theory which got a lot of experimental support of Emilio Del Giudice and Giuseppe Preparata. Now, self-organized water is not just some specific, though dynamic structure. So each vortex, it is the source of energy. Each tornado is the source of tremendous energy. Cloud may be the source of tremendous energy, and all this is self-organized water. So it is the source not of just energy, but of high density energy, which is much more important than dissipated energy of, say, heat. Now, from this point, why this is also important, may be important, and a little later we'll talk about this probably in more details. The energy is important for consciousness. And here I just remind you that brain consumes more than 20% of organisms' vital energy, constituting only 2% of living matter of the body. So, in order to think, in order to have the normal and alternative consciousness, we need to use tremendous quantity of energy. The source of this energy usually is considered to be only ATP, but later on I'll show that water itself may be the source of energy. I do not have time to talk about different ways of energy production by water. One of them, which we studied in much more details, is water oxidation. And oxidation of any fuel by oxygen we call burning. And burning is a source of high-density energy. And we have shown that if we take bicarbonate solution, carbonates are the necessary component of any living matter, bicarbonate in particular, they are ubiquitous natural catalysts of water oxidation. We have shown that bicarbonate supports stable long-term water burning. Later on, I'll show the equation of this water burning by oxygen. In particular, if you take simple just bicarbonate solution like just usual drinking water, hermetically close it and activate it, adding to it the active, a very small quantity of active oxygen in form of hydrogen peroxide, this water will start to emit photons and it will inflame more and more after just its preparation for many months. And in some cases, we observed this water burning for a couple of years already, even in complete darkness. So this water really burns. Now this burning water, we measure continuously this photon emission from this bicarbonate solution, just drinking water, in fact, solution, using the photon very sensitive photomultipliers and monitoring it for a long period of times, we could see that subtle energy irritations from environment affect properties of this burning water. Such irritation as moon and sun eclipses, moon cycles, geomagnetic storms, and here are three dots which by these three dots, I hide the effect of Hellas upon this burning water. And we demonstrated also that Hela may be at a distance of 2,000 kilometers from the preparation, which is standing in the photomultiplier, before the photomultiplier tube, and he may affect, increase or decrease this photonic emission from water. Here is the one of the examples of effect of moon eclipse on water burning. On the left, this is photon intensity of photon emission from the test tube. This is just average photon emission for a long period, rather long period of time. And here moon eclipse starts. And you can see that water becomes much more inflammable for many times, at least 2-3 times more inflammable during moon eclipse and even after moon eclipse, because it ended here at this time point. For 2 days after moon eclipse, water is burning very brightly. Then it returns back to its initial state, but there are some photon emission impulses, increasing photon emission even after moon eclipses. Same with sun eclipses, same happens during moon cycles, and so on. So water, this just normal water, bicarbonate water, which is the material basis of living matter, is very sensitive to all these very subtle influences, which in fact we may call informational influences. So why can water burn in principle? Because water is considered, of course, to be the extinguisher of fire and not the fuel. It is so specific that it is a fuel as well as it is an extinguisher of fire. As a matter of fact, this is the picture of real burning of water under the action of radio waves. This burning of water was, we can see this temperature of this flame is 1,500 degrees. It was discovered by John Cosnciousness in Pennsylvania about 15 years ago. Now, why water can burn? Max already told about the works of Gerald Pollack, of Easy Water of Gerald Pollack. We are talking, Gerald Pollack found that under not very specific conditions, water may turn from bulk water, which we usually consider in all textbooks, to very highly organized water, which he called exclusion zone water, easy water, which is water forms near the hydrophilic surfaces, and hydrophilic surfaces are the major surfaces in the living matter. Practically, if you look into a cell, say, let's take erythrocyte, then we calculate how much water is in erythrocytes. Average, it is 80% of water in erythrocytes and 20% of hemoglobin. But if we take in consideration dimension of hemoglobin molecules, then between adjacent hemoglobin molecules, there will be no more than 2 to 4 monolayers of water. Water like this. So this water which should be constrained, and there is in any living cell a very small quantity of water which we usually consider in glasses, in just our everyday life, which is bulk or chaotic water. So it was discovered by Pollack, but it was the state of such water was predicted first by Nasonov in 1930s, Troshin, his follower, by Gilbert Link and Emilio Del Giudice. So there is a lot of this exclusion zone water. And Pollack's achievement is that he studied very in many details the difference in properties of this constrained water which forms near the hydrophilic surfaces, and he has found that this water is surprisingly, it is charged. So if you put here an electrode here and electrode here, there will be electrical current between these two electrodes. That means that this water is rich in quasi-free electrons. And here I would like to remind you a little that I told a little earlier, that water in microtubulars has practically superconductive properties because these electrons should be very easily freed from that water. Now it turns out that this easy water may have much higher electrical conductivity than usual bulk water, in which we have this excess of protons, which are not very good the ions for transfer of electricity. As soon as this water is the source of electrons, it may be oxidized. These electrodes can bind to oxygen. That is, oxygen is reduced and water is oxidized. So the product of this oxidation of water is the source of energy. So and carbonates, as we have shown, are catalysts of this phenomenon. This is a unique chemical reaction, really unique. There is no other chemical reaction of oxidation of other reactions like this. Unique property of this process that products of water oxidation, here are the substrates of water oxidation, water which is fuel, oxygen which is oxidant, and products are again oxygen and water. There is no such a chemical reaction whose products and reagents are the same, but they are different in their structural properties, because this is highly organized water and this is more chaotic water. So free energy which is released is released due to increase in entropy. In fact, I don't have now opportunity to tell that this reaction is normally reversible. So in fact, it is not just burning of water, but under natural, normal conditions, natural conditions, which exist everywhere, there is a return of this chaotic water. They can be again transformed into the coherent, into constrained water. Now, just to go a little further, because we're already I'm probably should go to the end. I would like to give a brief summary of unique.
Vladimir, if you like to continue, that's fine.
Yeah, okay, I'll try then, because I need to tie up these parts again, water and consciousness. Now, I'll present brief summary of unique properties of aqueous systems. First, aqueous systems are capable, and not just capable, they continuously self-organized into holistic units. They may extract low density. In fact, this low density energy is infrared energy, and probably even much lower density. It may be far infrared energy, it may be mechanical energy, and so on, from the environment, and convert it into high density free energy. High density free energy is at least electronic excitation, but now I will not go deeper into this problem. Now it has been demonstrated that mechanical treatment of water may induce nuclear reactions in water. And this mechanical treatment of water is in nature, it goes continuously. Now they also that these holistic units may solve the source of coherent photonic or phononic radiation. And all these properties allocate them with the capacity to receive resonance, electromagnetic and other oscillatory impulses from the environment, and actively react to them, changing the properties of the structured processes which proceed in water. Now let's return again to brain. First, brain is one of the most wet organs of the organism. Brain is the most morphologically complex living matter. In all the universe, as we know, compact, not only complex living matter, but very complex and compact living matter. Water in brain is most sophisticatedly dynamically structured. Because if we look at the fibrous, at a brain as a fibrous tissue full of different. Well, everybody knows at least the basics of neuro anatomy. And all these axons, all these astrocytes and other cells, they have tremendously developed surface area, which are covered with this organized water. For example, so it is most sophisticatedly dynamically structured. For example, about this magnetic resonance imaging data, super organized water in microtubules, super organized water in neurons, also it should be, and around them. So, and brain is also the most intensely energy processing living matter. Of this I already told. The data that brain not just uses energy in the form of ATP and so on, but it uses energy of very high density, was first demonstrated by Alexander Gurevich in 1920s, 1930s. He discovered, I will not go into details, people, I suppose, heard about this, mitogenetic radiation, that is low intensity ultraviolet photon emission from living matter and aqueous system, which is indication of its highly non-equilibrium state. And he and his daughter Anna Gurevich, they demonstrated that brain in situ, or brain on the living, which is analyzed, for example, on living mice, is the most intense and continuous source of coherent, that is already according to Fritz Popp, ultraviolet photon emission among other tissues. Intensity of mitogenetic radiation or ultraviolet photon emission from brain exceeds intensity of photon emission from other tissues, sometimes 2 orders of magnitude. They also demonstrated the parameters for photon emission from brain are very sensitive to physiological stimuli, such as visual perceptions. And now these parameters of water were demonstrated their sensitivity to visual perceptions using functional magnetic resonance imaging. And also Gurevich insisted that brain is an excited continuum rather than collection of cells. And in fact, using magnetic resonance imaging, we see that brain is maybe considered by analogy is either the archipelago, or better to say the different lakes which are more or less connected to each other, to a sea, which after LSD treatment, it behaves like the whole sea. But these are results of 1930s using a very controversial, in the opinion of many scientists, technique, measurement of mitogenetic radiation. Now, already in our time, visualization of photon emission or biophoton emission can be made using much more stable and technique. And the Japanese physicist Kobayashi, with whom we work together with Fritz Popp, he using very sensitive photomultipliers has demonstrated that from a rat's brain in situ there is rather strong photon emission. And under occlusion of blood flow into brain, photon emission intensity fades. Sometime if you just then open again blood flow, then it returns back. But if it takes some time, if after a long delay of occlusion of blood flow, it doesn't return back. So that's in fact the explanation probably, or indication why halt in breathing for several minutes already is deadly, though from the biochemical point of view nothing should change in living matter after 5-10 minutes without oxygen. But in brain, the fire in brain fades, and there are maybe means to return it back, but not we don't know many of them. Coming back to consciousness, according to Giulio Tononi, one of the distinguished psychologists, neurobiologists of our time, a system demonstrating consciousness must have two specific traits. First, system must be able to store and process large amount of information. Consciousness is essentially a phenomenon of information. Second, this information must be integrated in unified whole, so it is impossible to divide it into independent parts. So consciousness is integrated information. I think that Marx already was talking about the first principle of water in conjunction with the DNA. So the water which hydrates DNA together with DNA is able to store and process large amounts of information. But I would like to add that without DNA, water may also store and process large amounts of information. I refer to data of Benveniste, of Luc Montagnier, and to many other scientists who studied the phenomena related to what we call with a bad word homeopathy. So another professor of psychology, Max Vermans, also wrote such a thing, that consciousness in some primal form did not emerge at any particular stage of evolution. Rather, it was there from the beginning. Its emergence with the birth of universe is neither more nor less mysterious than the emergence of matter, energy, space, and time. And I add to this, it is neither more nor less mysterious than emergence of water and of life, which is impossible without water. I just claim, that's my belief, my fantasy, that water is just the essence of living state, and as soon as water appears, and we don't now know how it appears mysteriously like matter, energy, and space, life also appears. There is no origin of life from inert matter. Life is such the same, is in the same row of notions like matter, energy, and space. And finally, just we come now to the end. Water is the most abundant substance in the universe. It exists in a permanently excited state. It cannot be divided into independent parts. Therefore, it may be the material entity integrating the world on organic, inorganic, organic, and consciousness state of matter. And Marx was, I liked it very well. He was talking a lot of telepathy. in order to have telepathic contacts, we still need to have some channels for transmission of this information. And transmission of this information may be made by the use of what we call entanglement. But for entanglement, again, we need some carrier. And this carrier, from my point of view, that is my fantasy, it is water, which is everywhere in the universe. So thank you very much for your interest. And if I have time, I am ready to answer several questions. Thank you.
Thank you, Vladimir. Your talk was wonderful. I invite more questions first. Please, people, turn on your microphones and ask questions.
Vladimir.
Yeah.
What makes you think that the photons that are emitted from burning water are coherent?
That's I can refer to Fritz Popp studies.
Oh, okay, right.
Yeah.
Okay, right.
In the first place, you see, that is a great science, as you know, which was originated not by photons, by Fritz Popp, and he has demonstrated in principle that all practically ultra-weak photon emission, so-called, is coherent. Especially it is coherent when it is originated from the structured source. And water, which emits photons, is a structured source.
Yeah. Easy question.
So
thank you.
that, yeah. Mm-hmm.
Any more questions? Vladimir, I will start with a generic question. As an expert of water, you probably know everyone of importance making special live water, therapeutic water.
Living water.
What is your living water? Yeah, what's your favorite device or technology which produces good water?
Well, my favorite device of living water is bicarbonate. It should contain at least 5 millimolar of bicarbonate, just usual drinking water, and it should be potentized. But to say so, it should be succussed. It should be succussion. Yes, it is very important, in fact. But I am lazy and I forget to succuss a bottle before I drink it. Though, and we have shown it, that succussion of water, it increases this process of water burning dramatically. And this water burning, that is origination of free radical, that is water self-purification of water due to this process, due to cavitation which you induce in water. This succussed water may stay for several hours or sometimes even more. It depends now about other. So
and
that is
what's the mechanism? How does that happen?
The mechanism is when you succuss water, there is cavitation in water, there is water splitting into radicals, and it was demonstrated that after succussion there is a small increase of hydrogen peroxide in water, which can originate from hydroxyl radicals, and hydroxyl radicals appear from water splitting into hydrogen and HO free radical. And this process, it doesn't fade immediately, as just many people think. It continues for very long, for many hours. And this water, when these radicals recombine with each other, this is the source of photons, or energy of electronic excitation. Now, this is the source of energy, like it is sparks which we add to our body in order to inflame our bioenergetic activity. So that's what
Yes,
I
Mark.
mean.
Mark, go ahead.
Yes. Could you please comment? There is in Russia, they sell water which is called пограничная вода. They claim they have done a lot of research. It has some very special healing properties. Can you comment on this?
Yes, I was related to this project. First, I would not call this water. May I speak English or I should speak Russian? I had to do. I know very well the inventor of this water. First, as a matter of fact, the name "пограничная вода" or interfacial water. It was he just used my talks about exclusion zone water, interfacial water, and so on, and he called his water this. In fact, he has this water. Now he claims that he has another sort of water. But what he was dealing with is water which was frozen by frozen not in a refrigerator, but it was frozen by decreased, very fast decrease in atmospheric pressure. So it lost gases, and in the first place it lost oxygen. This water, he got such ice without oxygen. And then he used this frozen water. Then he melted it, and he electrolyzed it. This water, and this water should be bicarbonate water. That's what we demonstrated. This water, after such procedure, it begin to contain. It begin to absorb, to be non-transparent in ultraviolet region. That means that after these procedures, from bicarbonate, there appeared some very strong antioxidants in this water. And when we found it, this man, he decided to not to notice it, because it was not a water, it was a solution of a very biologically active substance, which should be studied, and we had some ideas about its property and so on. This a very strong antioxidant may be not only healing, but also maybe very dangerous. Probably, as you know, now the games with antioxidants now nearly stopped, because a lot of negative, a lot of negative effects from antioxidants. So, and he, yes, and he decided to call this water пограничная вода, interfacial water, though it is, in fact, it cannot be called this way. It is a very biologically active substance solution.
Okay, but can you comment more? Biologically active in what direction? Positive or negative or both?
Direction. All antioxidants, so-called, they are also oxidants. These substances may so we know very well about the negative effects of free radicals, reactive oxygen species, and so on. Now it becomes clear, there are other sides of the coin, that without them there is no life. And when I was talking about burning water, it is water which itself produces these free radicals, and these free radicals convert their chemical energy into the energy of electronic excitation. So when water doesn't, when this process in water are very non-intense, this water may be called dead water. What is life water is water in which this process normally, regularly, in good organization go. But excess of these processes, that is just like, you see, again, as we are talking about fire. Fire may be used for good purposes, and fire may extinguish what we use. So we should be, with these radicals, we should be very осторожными, very
Yeah, I see. Yeah, by the way, I was in Moscow about a year ago and I bought this water and I took it with me. I was recommended by somebody who's doing in this area.
I say
It was
that it
pretty
is
sweet.
very, yes, again, it is very biologically active. That means that if you use it in very small quantities and on a regular basis, it really may and should help. But
Okay.
if to use it just in big quantities, as a matter of fact, I myself suffered from this water. Because when we begin to study it, we didn't know all these processes. When you drink half a glass of such water or glasses of such water, it may be like a poison.
Okay. Yeah. Okay. Thank you very much.
Any more questions for Vladimir? Vladimir, I have tons more questions, but I think we should start our panel. We are 45 minutes behind the schedule. So the next speaker will have to, Sergey, can you wait for another 45 minutes? Your microphone is off. Sergey, включите микрофон.
Я не знаю, I don't know if I can do it because I have a doctor's appointment. It's pretty soon after my speech.
How much time do we have for our panel?
So my appointment is at 3 o'clock and I have for driving like half an hour. And so like so I'm here until 2:30 New York time.
Give me the difference. I don't remember the time.
Okay. So
how many minutes do you have until you go?
Let me see. Half an hour, 1.5. So I have basically 1.5 hours.
It sounds reasonable. So let's do like half an hour panel and then we'll invite you. So in half an hour you can start speaking. Is it good?
Okay. That should be fine. All
right. So I invite everybody to discussion. Does anybody have burning questions to start? So everybody, all the speakers are invited to answer.
Let's talk more about DNA and consciousness. No, we got the water and consciousness, but and my work with healers, changing DNA. Do we have any other experimental evidence or theories? And what you also talked about.
Glenn, your camera is turned off, but the question is great.
Yeah.
So yeah, we have water, consciousness, and DNA. And the question is, why do we need DNA in the body if water is conscious sufficiently already? There is an evidence that brain is not necessary for thinking. There are people with PhD level and or at least master's degree which don't have brain. They have like 5% of the brain.
Right.
Some sort of encephalopathy, something like that. And so brain is not necessary, but why do you need the body? Why do you need the DNA to have human consciousness? Why can't you just do water, pure water? So that's the main question. And invite people to speculate.
May I just add to this?
Please turn on the camera if you don't.
Yes, may just to your question.
Yes.
Yes, to your question. Why do we need Mercedes if we have a wheel?
Right. Let's be more specific in biophysics.
Yes,
What we really
yeah,
need.
yes, because without will, there is no Mercedes. Well, then, do you need any Mercedes? I mean, any other just very sophisticated? Do you need computer if you have what they call it, shorty in English, the abysses or something like this? Because you can, if you have sticks, you can calculate with sticks. You see?
Yes. Let's go into DNA actual biophysics. Why do we really need?
Yeah, from my point of view, may I try to answer your question? I just believe into, and as a matter of fact, I suppose that you also said about this, that consciousness is ubiquitous. It is practically everywhere. And some people even insist that each atom has a consciousness and things like this, but that's all, only philosophy. Let's talk about living things. As a matter of fact, the consciousness is a good English word, awareness. I don't know how to translate it with one word into Russian. Awareness.
Осознание.
Осознание, knowledge. Not сознание, but осознание. So you should be aware in order to plan, to think about future, to have memory and things like this, you see. And these all things, they have, even single cells have such properties, though they behave much better when they are in a company, when you are having to do with population and so on. And from what I would like to mention is the difference. What is the difference between human beings and all other animals? For example, such animals like even primates.
But they all have DNA.
All have DNA, yes, all have DNA. But still, we somehow understand that there is some difference between us and chimpanzee. Of course, sometimes chimpanzee may be behave much better than human beings, but in general, but in general, there is such a difference, though our DNA is very, very similar to each other. And there is such a parameter. This parameter is called, it was first claimed by Erwin Bauer. That is quantity of energy which is used by a representative particular species, divided per his mass and multiplied by the lifetime. So there is this parameter Erwin Bauer called the constant, oh, I forgot the name of physiologist now. So this parameter increases in the course of evolution. It is very small, this parameter, for unicellular, for worms, and so on, and then it increases. And when we compare this parameter, for example, for primates and human beings, for human beings, the quantity of energy which they operate during their life is 10 times higher than for chimpanzee. But do we need such energy? First question. Because primates, they live for many hundreds of thousands of years, at least even maybe longer than human beings. And we have 10 times more energy than they. And we use this energy for what human beings really do. Where from this energy, where to this energy goes for? It goes for our brain. That is the major difference in bioenergetics of bioenergetics general energy performance by human beings and all other animals.
Yeah, but what does DNA have to do with it?
The DNA is one of the organizers of this energy flow.
Okay, well, let me add to that.
It organizes. Now, how it organizes it, about this, ask marks. he said he has shown very beautiful pictures about the participation of DNA in practically all processes. Because what we call genome, yeah, that is the coding DNA. That is just the I will call it instrumental box of a master. I have such a comparison when we consider DNA. Marx has demonstrated that that only 4% of DNA codes for some proteins. Yes.
Right.
Yeah,
Right.
and so 96% codes for what? For information. Now, when you invite a master to repair some your device, he comes with his instruments. The 96% is his knowledge, and 4% is his instruments in his box. Without his instruments, he cannot do anything, but without the master, instruments can do nothing.
Okay, so I'd like to add to that and
And
say
to
that.
and I'm sorry, and just I would like to tie it
Okay.
together. And
Okay.
for this, you need tremendous quantity of energy. And human beings, they possess this excess of energy over what is needed for survival of them as animals. We
Okay, so
possess 10 times more energy, which is than is needed for our survival.
okay, so I want to just add to that, say it's not only the amount of energy, but it's the kind of energy.
Yes, of course. Yes, I completely agree with you. I completely
And the
agree.
kind of energy that
Yes,
distinguishes us from
yes,
other species is
exactly.
divine energy.
Exactly. Here we can talk about coherence, about phase matching, and things like this,
Right.
about quantum properties of energy.
Quantum
Yes,
energy
I
and all that,
completely
right.
agree with you, because
Right,
it's
right.
not. Yes, there is same quantity of energy when you take a barrel of petrol gas. Yes, you can
Right,
a
right.
match in this barrel, or you can put this gas into gasoline into automobile. Same quantity of energy will be released, but with
Right.
different effect.
And I would also propose that that DNA in higher species
Yes.
is known to exist as a toroid. Now, I don't know if lower
Mm.
species has toroidal DNA, but I actually proposed a model for the toroidal antenna model of DNA, suggesting that DNA can act, the inside a toroid is a catenoid. A catenoid is a wormhole, and a wormhole can allow higher dimensional energies into the body. We have that. I propose that a lower species do not.
That's a good idea, but there are that's a very good idea, very interesting idea. But I should first would like to remind you that the circular DNA is in bacteria.
Yeah. Well, that's not
Even in
toroidal.
birth. But
It's
yes, but this
also
the
in mitochondria.
sorry.
I said it's also in the mitochondria of our
Also.
cells.
Yeah, but there are probably different levels of toroidicity. Yes, that's the geometrical organization may be different.
Yeah.
Several
Okay.
different levels. Like circular DNA is, of course, very different from DNA of chromosomes.
Right. Yeah, and it probably has its own unique properties. So what we're talking about is the geometric shape of the of DNA. And Max, do you ever talk about that in your theories? Or did
Max, switch on your microphone.
I'm talking to myself. Yeah, all about structure. Yeah, the whole talk was about structure of chromatin. DNA is wrapped around nucleosomes. These are very orderly structures. Tetranucleosomes are pretty orderly. After that, it is a dance of something which we know is not periodic. It's not pure crystal. Water around DNA could be crystallic, but it's very complex. There is no simple structure in chromosomes. It's complex. I'm looking at this structure for the last 4 months on chromatin conformation data, and it's complex. It's it's loops, and it's called compartments and loops, which are organized in such a way that DNA loops and then multiple loops combined together in sort of a blob where it's all combines sequence specifically. So there is a sequence specific program how it combines. One of the methods of controlling DNA structure is by twisting it. There are enzymes that twist it, like, in old days there were phones with a twisted cable, and if you turn the receiver several times, then the cable creates multiple shapes depending on the level of twisting, super twisting. So I think that's one of the major controls how DNA functions. But sequence is the key, and transposons are the key, how it folds and dances.
So have anybody ran across any scientific articles saying that twisted DNA, the function of twisted DNA?
Mostly 50 years ago. After that, people became fascinated with pharmaceuticals, and they forgot about it.
What
Let
did
me
they say?
address the question of human-centeredness. Like, it's a mistake to think that brain is so important, that consciousness is in the brain. It's also is a mistake to think that consciousness is particular for humans. It's more like the other way
Right.
around.
Right.
Like, I think the smartest and most conscious part of the planet is the planet itself. Maybe next after that is ocean. The ocean, I think, is pretty smart and conscious. After that, I think mushroom mycelium, there is a mushroom network around the planet, and it's interconnected, and it has the properties of the brain, and it's much bigger and smarter than all of us together. Then there is forests and plants and forests behave as a united organism. So they are smarter than individual humans. And when you meditate and go on astral trips, you just join that consciousness and you realize that humans are not the most smart, they're most confused. Like, we are most disconnected from the consciousness. That's why we're asking such weird questions. Once we get there, like in tribes and in naturally living people, they are much more connected to universal consciousness. We are utterly disconnected. And this utter disconnection comes from our programming, cultural programming. As I say, there are levels of brainwashing. The highest level is a medical doctor, a little bit below that is PhD. So we are very strongly brainwashed and disconnected from natural living, from telepathy and psychic abilities, and from actually being connected to universal knowledge. I think most of the nature outside of us is much more connected than we are. And the secret of finding the answer is to lose your egotistic personality separateness and to reconnect back to the nature and universal consciousness. It's a lifelong practice, but many people in many cultures, they achieve that. Now, coming back to the idea of DNA, I think the key here is that life based on DNA is different from all other consciousness, is that experience of being pulled forward in time and making choices. So I think the key of DNA is grounding us in one period of time and pulling forward. So I published that paper. It's called How Schrodinger's Mice Weave Consciousness. And essentially, I show there that we have a system of multiple collapsing systems, like one of the fastest collapsing decollapsing systems is electrons. They're very small, the mass is little, and the oscillations are high frequency. So they constantly delocalize and localize. And this collapse of delocalization is what Heimerov calls, what does he call? There is a word for that. So the basis of consciousness, anyway. And then after that, there is protons, which are about 1,000 times more heavy. They're doing that slower, but they also delocalize and localize. When they delocalize, instead of doing the clouds, they do the wires, like proton wires. And that process goes bigger and bigger in scale. And we as organisms are collections of cells, and each cell is smart. Each nucleus is smart. It has DNA thinking and water thinking. But there is an illusion of being separate. So this human separateness is an illusion created by DNA program. And brain is still important. In lobotomy, that was in like over 50 years ago, there was lobotomy as treatment for madness. And when they disconnect part of the brain using surgical procedure, the person was still alive, but was barely conscious. They were able to do basic tasks, but didn't have long-term memory and didn't have normal coherence. They were like more like a vegetative state. So brain is important, and DNA programs the structure of the brain. That's sort of more or less clear through morphogenetic principles. But also, I think DNA directly participates in thinking. And the culture, I think, imprints on DNA structure. It's not proven, but I just intuitively believe our learning goes first through the brain and electronic patterns in the brain, and synapses and synaptic plasticity, neuron axonal structures and how they reconnect. But then after integration, it goes into chromatin structure. And I think in chromatin structure, we have a big part of our physical mind, our personal mind. And that's why medications and psychological active drugs affect not only neurons, but also the whole body. And the health of the whole body is directly participating in emotions and state of the mind and state of the individual consciousness. So there is a lot of layers, and DNA participates vibrationally and biochemically, and it's a program that dances and thinks by itself. Sheldrake, Rupert Sheldrake, is one of his main messages is that a lot of the mind comes from outside, and DNA provides only limited function. To me, it's bigger than limited, but still, I think DNA works as a gateway, as a portal, as a door to universal consciousness. And what it actually does is filters out a lot of knowledge to create a small human mind and to make it forget that it is part of the bigger whole. And this is, I think, is written in DNA by culture. So imagine the culture switching the chromatin structure to make us forget what we are. And awakening goes through unforgetting and remembering who we are.
I have some comment.
Yes.
So you're completely right in my view on Max, on that our consciousness is connected to other species consciousness, but I think all of living beings, especially humans, they have their own uniqueness, which is expressed in mathematical term singularity. And here we come to the ideas of German great mathematician and philosopher Leibniz, monadas. So each of us is a monada. So if we dissolve this monada like in some ocean, like we are losing our consciousness. So yes and no, we are connected to the common consciousness, but there is no such thing as common consciousness, universal consciousness. There is consciousness everywhere because there are different species, different uniqueness. But what we share, it's not in my view, it's not the consciousness. It is inner world. And we are seeing, you know, like when we study materialistic science or physical science, we are just seeing upper part of the iceberg, and we are not looking at the bottom part of the iceberg. And the upper part of the iceberg is consists of DNA. Here is the brain, neurons, atoms, everything that we can measure and we can work with in the lab. While the bottom part of the iceberg is the inner world, which nobody really sees, but everyone feels that it exists. And it is perfectly explained in contemporary theoretical physics, because, you know, like inner world. And actually, Rupert Sheldrake tells exactly the same thing. You know, when he talks about conscious universe. So we all share the inner world. So inner world is being described by quantum mechanical wave functions, while outer world is being described by the fields, and those are completely different mathematical and physical objects, and they behave different way. And what is a part of DNA? Its role? Yes, it projects the whole inner world into one point. You know, where we are, like where I am or you are. You have your DNA, I have my DNA. So it works like a gateway, but gateway to what? I don't think it's gateway to common consciousness, you know, in my view. I think it's a gateway between us and inner world. But then in inner world, we connect to everyone, every living being. And here, what is helpful we can have for us from DNA research, besides, you know, like staying away of any genetic modification, which if you understand, gets deeper look, I'm pretty sure we will see how hazardous it is, you know, for the world. But speaking out of DNA role for living being, without us trying to do anything with that, it does work as a gateway. And if you understand how inner world projects to outer world, how our thoughts, our plans, our virtual path transmits into real path, you know, we will, and it transmits back as imaging of the world. I think DNA plays a big role. I don't think it's only one. A big role in this whole projection, which is all united into one singularity we represent. So this is part of, if you talk about DNA, if you talk about brain, those are external parts of our minds of our consciousness.
So Sergey, how does the DNA do that?
So DNA does that via biological field. And this is one point and one direction that Max also making research in. Seeing, you know, like that DNA is a great candidate on the source of biological field, in the sense of Gurbich or in the sense of Rupert Sheldrake. Those like theories that stay a part of mainstream. You know, like not biological field in the mainstream sense, which is not field, but in real field. So once we talk about the biological field, then we talk about outer part, and then it goes all of the events happening with this field, and they project via singularity, they project into inner world. And when we research DNA, we research at singularity, how singularity is being constructed mathematically and in a physical theoretical point of view. And here in my own research, I figured out that DNA has properties of spin. What is spin of electron or spin of proton? So it can be described with all of the genetic algebras. It can be described in the same formalism that spin is. So spin plays a special role in this conversion between inner and outer world. And spin is a rotational structure also. So the rotation plays or twisting plays a central role in our lives, and we know that by that life is a cycle phenomena. And so we basically have some mosaic, you know, like, and DNA plays in significant role in this mosaic. It's significant part of it.
Thank you.
I just
Good
wanted to
answer.
welcome Richard Alan Miller. Hey, Richard, turn on your microphone.
May I just I say something, but maybe not so sophisticated like you, Max. Your theory was amazing, and Sergey and everyone who, I don't know if you hear me or not. I'm not sure. Where is
You
my
good? You
mic?
good?
Yes.
Yes. Oh, okay. So I just wanted to say that's amazing what I hear, and I'm very excited. But if we imagine that everything has a consciousness, from little cells, from little creatures, whatever, from something which we don't even know in different world, in multi-dimensional world, and everything is the process of evolution. So we touched base different topics and tried to put it in one big theory. And so if we're talking about DNA twist, which Max was talking, is if we're talking about what Glen was talking, toroidal, very well, developed evolutionary DNA, and not only DNA, because the whole world is like toroidal, the universe, everything, right? So what I'm trying to say is that if we dig down to the process of evolution, like just I will narrow down to whatever mitochondrial DNA, which was like it wasn't like a normal DNA, right? So it was like a symbiotic coexistence or whatever it is. It's one of the theories. Then we can tell that only when you have not you, when the cells or DNA or any organella or anything has a pretty high level of consciousness, which is set of mind, and we create this morphogenic field around us, right? And I'm talking about all elements, doesn't matter what, right and left torsion field, what also depends on the set of the mind, of the mood, of the pure intention, of the power of your intention, of the light which you emit. Okay, so we create our own kind of universe, and then we can make by our set of your mind and set of your morphogenic biofield or informational field, call it like you want, Ethereum field. We can make DNA, and not only DNA, water in the blood, lymph, whatever it is, crystals, which in our body, we can make it change the configuration, spin, twist, field. We can even probably, I don't know, it's just like I'm fantasizing, but I'm just trying to unite everything you were talking about in one picture, right? And in order to jump on a higher level vibrational being, call it like God, right? Because everything is a mess. And I agree with Sergey that it's mathematical and everything. And he was talking from the point also that when we look at the protein production in the cell, right? So the DNA, if you I was on a lecture of quantum computer. So which quantum computer created the process of protein production, and it was all calculated. What is the best way in nucleosome? How it's just laid down the DNA, RNA, everything. It's true, but the beginning, I'm very scattering minded, but I'm just trying to say the beginning is the intention, the program, so-called program, and program comes with a process of evolution for each species, for each molecule, from the point of origination, based on the environment. And I'm talking about intercellular environment, I'm talking about human environment, sound, smell, I mean, mostly, of course, sound and electromagnetic and other fields which we didn't open yet. But sorry, I just tried to give you some crazy thought.
Thanks. So you brought me to the statement that it's impossible to build a unified theory of consciousness. Impossible. From our perspective, with our language, in our space-time, it will be always self-contradictory. That's why I titled my last two chapters, I wrote them during the last year and a half, Imperfection as the Foundation of Life. And imperfection is key. It relates to uncertainty principle of Heisenberg and Schrodinger's wave function, that you cannot really collapse and build perfect picture of DNA and consciousness from this physical model. You always have to, you will come to realize that the whole nature is self-contradictory. If you grab one part, the second part will become illogical. If you fix the second part, the first part becomes illogical. So the only way to model it is to put self-contradiction as a basis of the model of the consciousness. All right, Rick, you wanted to say something. Microphone is off. You need to turn on the microphone. Rick, microphone, Rick. Your microphone is off. Yeah, go ahead.
I'm good now?
Yep.
Okay, thanks.
Hey,
I don't get out very often. I'm on. Please excuse me, Max, for not joining you much earlier, way back when. I'm being deployed right now by the military. Atlas.
Oh, congratulations.
You don't want to have any clue what's going on, but I know I don't know. But I can say this: consciousness is a dream within a dream. Lucid dreaming has more content to reality than consciousness. You're in stasis right now, and our future just entered our solar system. What does that mean? Good luck. And Max, when you get a moment, give me a call. I haven't talked to you for so long, and boy, am I telling you that I have no idea what I'm doing. And I know, look at this, I'm still deployed. At 81 years old, I'm a GS-18. That means I don't report to anybody above me, and the other two people in history, they're dead. That's when Max, it's been a deep honor to work with you, by the way.
We have a next speaker rushing to his doctor's appointment. I would like to thank Richard Alan Miller, one of the founders, the founder of DNA resonance and holography. Sergey Kotkovskiy will speak in Russian, and to turn on English subtitles in Zoom, in the bottom right, there is CC in a frame. Click on that, switch spoken language to Russian and caption language to English, and you will see translation. Sergey, you can start sharing your screen. And turn on your microphone. We can see the screen. Yep. Turn on the microphone if you did. Yep. And switch to this presentation. Yep. Perfect. Thank you. You have 40 minutes.
Okay.
10 minutes of that is question.
So for English-speaking participants, hopefully the translation will work great, but also visual representation, video, images, they will help us, nothing else. Мой доклад, он посвящен одному из самых загадочных и много изучаемых явлений в нашей жизни, который носит название Phyllotaxis и который заключается в формировании особых симметричных спиральных паттернов, когда растут растения. Мы это с детства знаем, мы видим их на подсолнухе, мы видим, как у нас цветы растут. И Phyllotaxis – это вездесущее явление, которое находится в центре вообще жизни как явления. И несмотря на то, что оно уже изучается более 250 лет, пока понимания его, почему это так происходит, пока его нету, и мы постараемся пролить свет на это. И увидеть, что в Phyllotaxis у нас имеет место фазовый переход. Итак, наш доклад состоит из 3 основных частей. Мы сначала обратимся к общему изучению, к общей картинке фазовых переходов, которую мы имеем в физике, и за ее пределами. Затем мы рассмотрим золотое сечение. И 3-я часть будет непосредственно посвящена Phyllotaxis и его представлению как фазового перехода 2-го рода. Итак, фазовые переходы. Фазовыми переходами физики называются переходы, которые резко меняют какое-то состояние. То есть у нас, допустим, был лед, температура понизилась до 0 градусов, и, то есть наоборот, повысилась, и лед растаял, получилась вода. То есть у нас переход между 2 фазами. Ну или там вода потом, соответственно, у нас мы ее нагрели до 100 градусов, она испарилась, у нас превратился пар, да, поменялось состояние. Но не все переходы носят характер изменения агрегатного состояния. У нас имеют место фазовые переходы 2-го рода, когда у нас агрегатное состояние не меняется, но и при этом у нас вся система, которую мы рассматриваем, она вдруг при непрерывном изменении параметров, она вдруг меняет качественным образом свои свойства. Ну, самыми известными примерами фазовых переходов 2-го рода являются ферромагнетизм, электричество, сверхтекучесть и сверхпроводимость. И давайте мы опишем немножко о том, какая есть у нас на настоящий момент теория фазовых переходов 2-го рода. Она была развита в 1937 году великим физиком Львом Давыдовичем Ландау. Вскоре после этого он сел в тюрьму, но потом его выпустили благодаря усилиям нашего другого великого физика Капицы. Ну, кстати, год 1937 тоже не случайен, поскольку в этот год, как мне кажется, произошел социальный фазовый переход в Советском Союзе, не в лучшую сторону. И, возвратимся к физике. Каждый фазовый переход 2-го рода характеризуется критической точкой, через которую он проходит, и когда это возникает, при ее прохождении возникает новый порядок, новая симметрия меняется, и то, что возникает, то, чего не было до этого, оно носит название параметра порядка. Но в случае ферромагнетизма это параметром порядка является намагниченность. То есть, если у нас температура понижается, то ферромагнитный материал, например, железо, он приобретает магнитные свойства. Но это не означает, что он сразу становится магнитом. Там его еще нужно будет намагничить. Но вот эти домены, которые уже у нас направлены, организованы определенным образом, они у нас имеют место. Как мы видим, при переходе ферромагнетики, ферромагнетика – это обычный магнит, по сути, железо точнее. А обычное железо – это у нас, которое можно превратить в магнит. При высоких температурах, выше точки Кюри, у нас все магнитные моменты на микроуровне, да, частиц, там, атомов, да, они и молекул, они направлены хаотично. Ну или это можно сказать, что здесь присутствует симметрия центрального вида. То есть если мы вокруг центра, она будет сбалансирована. У нас то, что сюда смотрит, оно смотрит и сюда. А при достижении точки Кюри, значит, в доменах у нас все они начинают выстраиваться строго определенным, спонтанно выбранным образом. А если у нас мы включим еще магнитное поле внешнее небольшое, тогда они выстроятся все по этому магнитному полю точно. Значит, главное свойство фазовых переходов второго рода, как показал Ландау, это изменение симметрии. Возникает из той симметрии или ее отсутствия возникает новая симметрия. Ну, кстати, сказать, что теория фазовых переходов второго рода, она не обязана быть строго внутри физики конденсированных состояний или вообще физики. В частности, моим учителем Щенниковым и его коллегой Белоцерковским, который очень известный ученый, был разработана теория кинематического фазового перехода. Это касается того, как вихри в жидкости организуются на микроуровне и объединяются на макроуровне. То есть у нас возникает общий фильтр, общий вихрь. Ну, теперь можно приступить уже к следующей теме, когда после предварительных замечаний о фазовых переходах, которые нам потребуются для дальнейшего. Да, мы сейчас приступим к рассмотрению золотого сечения. Золотое сечение, оно известно с древности, даже египетские пирамиды там находят золотое сечение в отношениях гипотенузы наклонной и катету. Древние греки точно знали, они умели строить пентаграмму, пятиугольник, основываясь на золотом сечении. Золотое сечение представляет собой вот такое число, и его главное свойство то, что там сумма, значит, составляющих треугольника относится, не треугольника, а отрезка, да, то есть длина отрезка относится к его больше составляющей, как больше составляющей, к меньше составляющей. Ну, то есть здесь какая-то рекурсия такая, вот такого типа присутствует. И эта рекурсия, она, оказывается, играет центральное значение практически во всех процессах роста, формирования организмов, ну, то, что называется морфогенезом. Ну и не только там, и даже в неживой природе. Что интересно, золотое сечение, оно является критическим для воспринимания красоты и правильных пропорций. Это еще было выяснено великим Леонардо да Винчи. То есть он это увидел, стал это использовать. Ну а само золотое сечение и числа Фибоначчи, которые с ним связаны, они пришли в Европу вместе с ее первым самым крупным математиком Леонардо Фибоначчи, или его еще называют Леонардо Пизанский. Да, и он открыл, изучая задачу о кроликах, которые размножаются, он открыл вот эти замечательные числа Фибоначчи, которые, как оказывается, связаны с золотым сечением. А вот их, значит, каждое последующее число равно сумме двух предыдущих. То есть 3, 1 плюс 2, 5, 2 плюс 3, ну и так далее. И вот, как оказалось впоследствии, числа Фибоначчи, согласно этим числам, строится процесс филотаксиса или процесс роста растений. Но также Сергеем Валентиновичем Петуховым были введены комплексные числа Фибоначчи. И вот как раз эти комплексные числа, они и описывают спирали филотаксиса, которые мы рассмотрим непосредственно в следующих слайдах. Также наша секция посвящена ДНК, и как это ни странно, в ДНК тоже работает золотое сечение. А может, это как раз не странно, а закономерно, если мы говорим о том, что золотое сечение является математической основой жизни, но оно, кстати, является математической основой и самих чисел. Оно, как по выражению Петухова, это проточисло. И оно является самым рациональным из всех иррациональных чисел, то есть его наиболее сложно приблизить, да, то есть как к нему приблизиться. И это свойство, то, что к нему наиболее сложно приблизиться с помощью рядов вот этих рациональных, оно как раз и легло в основу того, что в природе происходит, оптимальное распределение. Значит, в ДНК у нас есть 4, как известно, нуклеотида, и они обладают водородными связями. У каждого из них либо 3, либо 2, и из этих водородных связей можно представить золотую матрицу, и далее можно представить всю иерархию, значит, нуклеотидов, нуклеотидных цепочек. И эта иерархия именно в случае золотой матрицы, она становится, она превращается в некую инвариантно-мозаичную структуру. А сама она выглядит следующим образом, то есть вот эта матрица водородных связей, а это у нас золотая матрица, которая и представляет собой корень. Нами была открыта еще и вторая часть золотого сечения, комплексная. Ну, комплексные числа, как Глену тут уже рассказывал, они представляют собой на самом деле более фундаментальное числовое пространство, чем это делают вещественные числа. Как мы знаем, и квантовая механика описывается комплексными числами, так и электромагнитные поля, это для них самый натуральный, самый естественный язык описания, это комплексные числа. Ну, таким образом, мы увидели, что золотое сечение, оно имеет отношение к ДНК. Но самое главное в этом докладе это то, что золотое сечение, оно играет первостатейную роль, то есть на нем основывается явление филотаксиса. Значит, филотаксис, как мы уже заметили ранее, он занимается изучением паттернов, которые образуются листьями или другими органами растений, ну, в том числе с семенами. Да. Самое наглядное это у нас подсолнух. Да. В подсолнухе мы видим филотаксис, мы видим, что с детства мы знаем эту картинку, что у нас есть множество спиралей, которые в одну сторону и в другую сторону идут. Ну, вот этими спиралями мы сейчас и займемся. Значит, следует упомянуть, какие исследования проводились по филотаксису исторически, это уже более 250 лет. Начиналось все еще из Кеплера, по-видимому, он первый заметил, что в филотаксисе у нас участвуют числа Фибоначчи. Занимался великий Гёте. В начале 19 столетия братья Браве вывели ряд теорем и представлений о спиралях филотаксиса. Затем уже в начале 20 века Гурвиц доказал свою теорему о приближении рациональных чисел, приближении золотого сечения рациональными числами, показал, что золотое сечение действительно играет выделенную роль для чисел. То есть, если можно, в принципе, сказать, что все вещественные числа являются, строятся на основе золотого сечения. Не на основе натурального ряда, а именно золотого сечения. Ну и мы здесь займемся алгоритмом Фогеля, немецкого исследователя, который очень удачно описал филотаксис. А само золотое сечение представляется вот такой непрерывной дробью со всеми единицами. И как это показывается в математической биологии, занимающейся изучением филотаксиса, именно вот это вот распределение такое, оно и отвечает за то, почему филотаксис идет по неким правилам, связанным с золотым сечением или золотым углом. Сразу забежим вперед и заметим, что золотой угол – это у нас угол, который вот определяется золотым сечением, фи, и он равен такой величине 137 градусов. И значит, если у нас будут появляться семена или цветочки, листики, то вот здесь будет первое. Вернемся назад. Здесь дальше следующее у нас повернется на этот угол и так далее. Вот они все будут по кругу идти, но не совсем по кругу, поскольку у нас они должны дальше занимать места, и они пойдут, значит, у нас по спирали. Но прежде чем мы перейдем к самому алгоритму того, как это происходит, алгоритму представления, да, следует обратиться к тем системным исследованиям, которые были сделаны в Phyllotaxis. Это целая большая наука, и огромный труд был написан Джаном, и он есть в переводе на русский язык. Вот здесь системное исследование морфогенеза растений. Сергей Валентинович Петухов принимал участие в этом издании, и сам Петухов внес свой вклад в развитие Phyllotaxis в плане связи его с морфорезонансным полем, с резонансами. Почему оно связано с резонансами? Как мы видели выше, у нас есть матрицы золотые, да, по которым можно этот Phyllotaxis тоже с помощью них его представлять. Но эти же матрицы, они как раз играют роль и в механике, связанной с резонансами. Соответственно, у нас имеет место какие-то в этом явлении резонансы, которые мы пока еще не научились видеть. Да? Но мы уже видим их математически. И это очень важно. Также в Phyllotaxis многие ученые, некоторые ученые определили, среди них Ридли, Ревье, определили существование некого вариационного принципа. Это означает, что Phyllotaxis, он дает определенную оптимизацию. И у нас возникает однородность вот этих паттернов, и благодаря вот этой однородности, то есть они там не сужаются, там они не вырождаются, у нас происходит некая оптимизация. Также Phyllotaxis выходит далеко за области ботаники или биологической математики, в область неживой материи, так называемой, это квазикристаллы, полимеры, белки, вирусы, медузы, даже облака и даже упомянутые в предыдущих докладах микротрубочки мозга, которые Пенроуз связывал с самим явлением сознания. Хотя мы не согласны с тем, что явление сознания сводится к мозгу, да, является продуктом деятельности мозга, скорее наоборот. Но, тем не менее, мозг и микротрубочки действительно играют большую часть в самом этом явлении. Ну и последнее, что это Phyllotaxis, он представляет собой процесс упорядочения материи, а значит некого потока негентропии, да, создания порядка из хаоса. Значит, Phyllotaxis развивается по правилу золотого угла, и когда мы это правило используем и вводим еще некое правило, как у нас будет по радиусу это развиваться, то мы получаем известную спираль Ферма. Ее в Phyllotaxis, там своя существует терминология, вот эти семена, они называются примордиями, угол вот этот, по которому идет появление примордий, он называется углом расхождения, а вот спирали, которые у нас появляются в результате неожиданно, да, как некий, любят сейчас говорить, эмерджентный эффект, они у нас называются парастихи, то есть боковые спирали по-гречески. Ну и вот этот алгоритм немца Фогеля, он оказался настолько удачный, что полностью лег, на него ложатся подсолнухи. И каким образом он его выбрал? С тем, чтобы у нас вот эти вот участки, которые формируются, новые точки, да, они у нас занимали примерно одинаковую площадь. Они все совершенно разные по своему виду. Ну вот эта равная площадь, она сохраняется, хоть мы уйдем там на 10 километров. То есть получается совершенно такая однородная в плане общего доступа к Солнцу, вот этих точек, к дождю структура, но в то же время она совершенно не монотонная, то есть у нас она не вырождается. И это тоже чрезвычайно важное обстоятельство. И, значит, мы воспользовались этим алгоритмом Фогеля и построили вот эту систему, ну, мы не первые, конечно, да, вот эту систему, значит, спиралей, которые возникли, точнее, мы ее не строили, они возникают сами. То есть это такой процесс эмерджентный, да, можно здесь этот термин использовать. И этот процесс, он оптимизирует, вот это мы говорили выше о принципе, вариационном принципе оптимизации или минимума, максимума. Он оптимизирует доступ каждого из цветков, каждого из листьев к солнечному свету, к дождю, к воде. Именно потому, что каждая из этих точек, да, она занимает, которые соответствуют семенам, она занимает одну и ту же площадь. Вот так вот все достаточно просто. Но простота это здесь нам только кажется, поскольку процесс внутри он достаточно сложный. И здесь мы не видим, как это происходит. То есть мы видим, что это происходит, но как и почему. Следует отметить то, что золотой угол выполняется, то есть он должен быть взят с необыкновенной точностью. Если мы чуть-чуть эту точность изменим, угол, даже вот с помощью вот этих приближений фибоначчиевыми числами, то у нас все разрушится. У нас вместо вот такой картинки однородной у нас появятся спирали, которые направлены влево или направлены вправо. Вот у нас, значит, в нашем алгоритме мы получили вот такие вот, ну, аналогичную картинку. Да. И когда у нас угол изменяется, то здесь угол меньше золотого угла, здесь угол больше золотого угла, а здесь угол золотой. Вот мы налицо здесь изменение симметрии. Да, ну давайте немножко сейчас прокрутим видео, которое нам покажет, как это все у нас меняется. Так, сейчас. Да, вот оно поехало от малых углов. Вот здесь оно что-то начинает. Вот здесь уже схватилось. Вот, нет, здесь оно схватилось. А здесь оно пошло уже дальше раскручиваться. Да, ну то есть мы видим, что спирали превращаются в некую двойную спираль, да, подобно тому, как у нас есть двойная спираль ДНК, вложенная друг в друга. У нас здесь система, семейство двойных спиралей. Здесь уже пошла единичная спираль. Да. То есть у нас единичная спираль, которая имеет, давайте еще раз ее посмотрим. Вот здесь вот в этих, вот эта точка, да, эта точка подобная, сложно ее словить, конечно, поскольку она очень точно должна выполняться. То есть когда она непрерывно видно. Мы непрерывно изменяем параметр угол, и у нас скачком изменяется симметрия в этой точке. И симметрия изменяется от, значит, спиралей, которые направлены влево, к спиралям, которые направлены в обе стороны. Да. А если мы превысим угол, у нас, значит, спирали будут повернуты вправо. Значит, Phyllotaxis, он создает картинку вот этих паттернов. Во-первых, он создает оптимальное распределение. Мы видим, что здесь у нас никаких оптимальных нет. У нас вообще куча свободных мест, пустых. Да, то есть здесь в глубине может быть можно сделать как-то больший доступ, да, для каждого из них. Точнее, лучшее распределение. Но в целом, чтобы картинка работала, нам именно нужен этот точный угол. Кстати, этот угол, он не может быть, это подметили вот исследователи, значит, у нас как раз Ридли, да, и Прусенкевич, они подметили, что этот угол, он не может быть полностью храниться в ДНК, например, генетической информации, поскольку природа, она так точно не оперирует с величинами, да, то есть если он начнет нарушаться, то у нас все разрушится. У нас никаких ни цветов, ни деревьев, ничего этого не будет, поскольку все выродится, в конце концов. Все листья там на том же самом дереве, если они будут распределены неоптимальным образом, то это все быстренько засохнет и завянет. Да. А при золотом сечении все у нас золотое, да, у нас все живет и цветет. И это, кстати, большой аргумент в пользу того, что мы не являемся запрограммированными машинами, да, живые механизмы, как это принято считать, что вот ДНК это некая программа, да, по которой выполняется. Ну вот этот, если глубже вот вникнуть в природу филотаксиса, то он показывает, что здесь алгоритм просто сам по себе, он не может работать. Он может работать, когда мы на компьютере это делаем, а в природе это не сработает. Итак, мы описали филотаксис как фазовый переход второго рода, в котором управляющий параметр, когда у нас термодинамика это температура, у нас точка Кюри, то здесь у нас угол расхождения, так называемый, по которому он крутится. И именно в точке перехода этот угол равен золотому углу. Параметры порядка, которые, то есть что у нас возникает в результате филотаксиса как фазового перехода, у нас возникает вот эта структура спиралей. Наличие встречных спиралей, направленных вправо и влево, которые можно тоже назвать двойной спиралью. И у нас возникает однородное распределение, которого не было доселе. Значит, мы забыли сказать, что математически, так, где же она у нас здесь? Математически спиральные паттерны, которые у нас здесь, они образуют новый тип симметрии, подобный, например, тому, который у нас симметрия относительно вращения, например, цилиндрического. Да? И это математически строго доказывается вот в этой работе. Жан-Поль, он это показал как раз на основе теоремы, на основе представления о Браве цилиндрического, еще братьев Браве, которые были в 19 веке, работали. Да, и он показал, что это новый тип симметрии, который можно изучать аналогично тому, как изучают обычные типы симметрии. Ну, там, например, там Лоренцево преобразование там, в физике, да, или симметрии кристаллов. Вот физика конденсированного света, она имеет дело с кристаллами, да, симметриями. А здесь мы имеем дело тоже что-то похожее на кристалл, да, по крайней мере, структура у него есть, да, и у него есть своя математическая симметрия, которая именно приобретается в результате вот этого перехода, который мы, как мы считаем, он является фазовым по всем характеристикам. Ну, еще одно замечание, что в результате этого, как можно было видеть по картинке, когда оно шло, у нас периферия, да, как бы, и центр, они объединяются в одно целое. То есть, если у нас, вот здесь у нас центр, он в одну сторону закручен, периферия в другую сторону, и они совершенно разные, то потом они сходятся вместе и образуются одно целое. То есть, что такое филотаксис? Это создание целого единого, и наши природные объекты, мы сами, растения, животные, мы все являемся целым. Да? В нас, это еще предмет исследования будущего, я думаю, что подобные же механизмы, они лежат в основе морфогенеза, там, соответственно, создание наших тканей из клеток. Там тоже уже золотое сечение, уже есть исследование, что оно там появляется. И мы сделаем следующую гипотезу. При фазовом переходе филотаксиса возникает макроскопическое биологическое поле. То есть биологическое поле вот этого соцветия всего, да, или корзинки подсолнуха, да, но всего, по сути, это подсолнух, его корзинка, она формирует. Да, то есть оно вырастает из каким-то, ну, я думаю, что там биологическое поле, оно есть и до этого, но именно оно обретает целое, единое целое, макроскопически подобно тому, как макроскопически выраженное, подобно тому, как магнит у нас становится полностью магнитом в ферромагнетике в результате фазового перехода точки Кюри. Также у нас это происходит в растениях. Но это не означает, что у нас во времени оно происходит И растение, оно всегда с момента своего рождения, как и животные, да, они все обладают биологическим полем, макроскопическим причем. И вот этот базовый, он происходит не во времени, а в неком ином пространстве, да, вот пространстве углов, которые мы рассмотрели, да, и как бы мы его не можем фиксировать, например, как мы это делаем в случае ферромагнетиков, когда мы там, допустим, будем охлаждать, у нас появились магнитные свойства, да, а нагрели они у нас пропали. То есть мы такого рода эксперименты мы сделать не сможем. Но это не означает, что мы не имеем дела с подобного же рода феноменом. Да, то есть это биологическое поле, оно, как это и видел и Гурбич, и другие исследования, оно во многом подобно магнитному полю, и его наличие, оно является результатом фазового перехода второго рода, согласно нашим представлениям. Итак, сделаем спирали по растению, вот эти структуры, которые мы рассматривали, они строятся не последовательно, а возникают в результате процесса построения всего целого. То есть мы специально вот эти спирали, и они специально не строятся, а вдруг они возникают. Да? То есть они характеризуют вот эту общую симметрию живого организма. Phyllotaxis обладает всеми характеристиками фазового перехода второго рода. В результате у нас этого перехода возникает макроскопическое биологическое поле. Сам этот Phyllotaxis, он не является генетически запрограммированным процессом, хотя наверняка там ДНК играет роль в этом всем, но не как исполняемый алгоритм. Значит, отсюда это является также одним из аргументов в пользу того, что живое не является механизмом, как сейчас это принято считать в мейнстримовской науке, что живые организмы, живые существа, они являются биологическими механизмами, да, то есть основанными на работе на биохимических процессах. Значит, что важно для нашей человеческой деятельности, то, что Phyllotaxis, он не вырождается, и в то же время он является циклическим процессом. То есть там у нас все время что-то новое получается. Там, если поизучать вот эти паттерны, там целая наука, там у нас никогда оно не зациклится само на себя. И в то же время это циклический процесс. Ну, мы знаем, что наша жизнь представляет собой циклический процесс. У нас каждый день начинается с того же самого, да, и вот оно нам может дать некие хинты, некие советы по тому, как сделать и нашу жизнь, чтобы она не превращалась в монотонное существование, несмотря на наличие, или благодаря наличию циклов, чтобы она не вырождалась, а наоборот развивалась. Это я бы назвал цельной системой действий по принципу Phyllotaxis или золотого сечения. Ну и можно дальше перейти к некому обобщению методом индукции, а правильно сказать методом рекурсии. И это обобщение заключается в том, что и сама жизнь вслед за Phyllotaxis является фазовым переходом второго рода, то есть созданием организма как целого, не сборки его по отдельным частям последовательной, как это на конвейере происходит в цехах, а что она возникает сразу целиком. Но мы можем визуально увидеть, что у нас здесь что-то собирается, там что-то собирается, но оно собирается все не последовательно, как бы на линии, да, оно собирается как некий общий единый паттерн, значит, картиной, по которой оно собирается, чертежом, является вот это биополе, о котором мы говорили, биологическое поле. Да, ну, значит, вот это главный вывод такой, да, про нашу жизнь, да, про то, что Щенников называл, Владимир Щенников, он называл это процессом оживления неживой материи, да, то есть чем отличается живая от неживой, ну, там принципиального различия, если вглубь пойти, нету, но вот в тех формах, которыми мы имеем место, то мы видим, что живые организмы, они принципиально отличаются от неживых объектов, да. Происходит некий там что-то кардинальным образом у них другое, да. Но в неживых объектах мы тоже находим какие-то знаки внутри, в глубине, мы тоже находим жизнь, да. То есть мы все равно найдем там те же самые атомы, молекулы, да, и какие-то другие микроорганизмы, да. То есть мы все равно, у нас жизнь присутствует всюду, и это аргумент в пользу того, что называется единой сознательной Вселенной. Да. То есть Вселенная, как я говорил ранее, на мой взгляд, это не она сама обладает сознанием, а она является средой сознательных существ на всех ее уровнях. Да, ну, на этом я бы закончил. Да, если будут какие-то вопросы или комментарии. Сейчас, да? Так, я не слышу и не вижу почему-то ничего.
I asked Glenn to co-chair, but I think he
I'm
got.
here. But he's speaking in Russian. I don't know if it's the end.
Oh, that was the
Okay.
time for questions. Go ahead, Glenn. Glenn is co-chairing now. Glenn, you're not heard. Maybe your microphone is off.
No, I'm here.
Yeah. So you can invite the questions.
Yes.
У меня есть один короткий вопрос.
Yes,
I have one short question to Sergey. Sergey, you just mentioned in English, right? Should I ask in
English is fine, yeah. I just
So I was just wondering, because I was toying with the idea about like collective consciousness mind and how it's dramatically changed for the last 5 years. I'm talking about humans. So so what about the universe, which you said it doesn't have their own consciousness, the whole universe, but it has like individual, I mean, collective consciousness of kind of other consciousness field of different species, like collective consciousness. So it could be religious, it could be, it's not only human, it's like animals. What do you think? So is it possible to have a mathematical equation or like a tool to change the reality by the shift of collective consciousness mind without scopolamine or like whatever mind changing medicine or something like that?
So you talk about the ways how to change our consciousness state without any substances.
Yeah, just to make our life better. I'm just trying to look at the global, the big picture. How can we change the situation? And I'm talking about from molecular level to human consciousness level, based on what you said about like, because the universe is like it's like integration and combination of many different levels, and I would say even dimensions.
Yeah, we have like Max said, like he thinks that the ocean is its own living being, like which I basically agree, but I think it's not only ocean, it's whole biosphere, whole planet is a living being. So we talk about like different level, like Earth, the whole Earth is a living being having its own mind, and we kind of like it gives us a shelter to live. Yeah, it doesn't mean that we are somehow we are part of this mind, like similar, like microbes, probably part of our organism. But talking about how we can enhance and develop our consciousness without any substances, about like what is called change state, because like what substances do, they basically destroy our neurons, and like this is very hazardous and self-destroying way. So we need to find something better than that. And that's probably one of the directions, not probably, but definitely one of the directions where this research is one of its goals. And when we work with our own consciousness, we work with and we may try to connect internally, like what we do in our night dream, to whole inner space and find other consciousness. So we may develop some common field, like now we have 6 or 7 people sitting together remotely, and we kind of generate common consciousness field, conscious field. But it's not like organism. It's different. It's like it's temporary. While organisms, they quite like at least like humans, like we believe they're eternal. Yeah, even after death, like our mind is not being, it doesn't disappear. So we talk about 2 things, like when we talk about collective consciousness, and we talk about individual consciousness, and real consciousness is individual consciousness. That what is represented by singularity. Singularity is some kind of uniqueness in the whole universe. And when we talk about collective consciousness, we talk about some phenomena that is created for a while, but which is really great, that we can combine our minds together and create it. But it's not actual consciousness. And it is often being confused, and now we talk, especially with AI and with neural networks, with noospheric research, there is a huge confusion, and the people say, okay, let us combine our minds, and we will dissolve in this, and we will create one common mind. And I think this approach is very dangerous, like
I agree.
to the civilization, because we may dissolve as individuals and become parts of one big quasi-organism, like which. So that's something that we need to think how to prevent it. Yeah.
Right. Yeah, but how to prevent it, it's one thing, how to change is another. Have you, maybe somebody here among listeners, have you read the book of Monroe? He was talking about some specific, I forgot it, like 3.7 hertz or whatever, like very pretty low frequency, when the mind is shifted so beautifully, and you can just get out from the body. So it's basically what I'm trying to say. I haven't tried, but maybe by the special conditions, by not only sound, but many different conditions, we can make this new field or something. Put the glasses, like eyeglasses, press the buttons, and it will be like the program. I mean, I'm trying to.
Well, we have Irene's tuning bowls on top of her head. That's why I asked her what frequency might facilitate such a transition.
Yeah, it's different. Yeah, the tuning bowls, it's individual. She changed her. She's just.
Yeah, one by one. Yeah, you want a frequency that we want to broadcast throughout the whole planet.
Yes,
exactly.
Exact.
Okay.
Exact.
Right. Well, I would use the frequency of the crown chakra, which may or not be real. The frequency, we don't really
But
know.
it has, I guess it has to go in conjugation with the math, with a sacred geometry, with a golden ratio, with what Sergey was talking, which is amazing. I didn't know a lot what he just presented, and I'm fascinated. And so if we combine everything together, what we know, yeah, I'm sorry, I usually don't talk too much. I don't
But
know
we
what.
have to let Sergey go to his next appointment. So it's been 45 minutes and we should move
So
on to
thank
the
you,
next
Sergey.
speaker. So,
Okay,
Sergey,
thank you.
thank you very much.
Yeah, I hope it made sense for you, like my talk, and you will once you go to the slides, you will get a lot of understanding, in spite of it was in Russian.
Okay, great. Good job. Well done, everyone. And thanks everyone for hanging in here for the last speaker. We're going to hear from Mark. Mark, are you ready?
Yes, I am.
Okay. Well, Mark's going to talk to us about more mathematics of remote viewing. So we're back to raising our consciousness and getting out of our physical four-dimensional brain.
Yeah, actually, I'm a mathematician. I'm using mathematical approach, but it's the talk is completely non-technical.
Non-technical. Okay, great.
Completely.
Good.
The approach is yes.
Well, thank you very much
in the
for
spirit
here.
of applied mathematics, but completely non-technical.
Okay, so if you're ready,
Yes.
put up your slides and
I am.
do the share screen.
Okay, here are my slides, and I need to share screen, right?
Share in the bottom.
Yes. I share screen. Okay. Can you see? Yeah, can I make it full screen somehow? Can you see my slides or not?
Yeah, I can see it.
To do full screen, yeah, to the left
Yeah,
of that thingy, just
that on the bottom, just to the left of the
slider, to the left of the slider.
Yeah, slider. Yeah.
Lower, lower,
Okay.
Lower.
Right bottom corner, внизу около движка, еще левее, чуть-чуть левее, чуть-чуть левее. Там движок, и слева от движка такой экран стоит на палочке. О, F5, кнопка F5 на клавиатуре. The button F5. On the screen at the bottom of the PowerPoint, there is
to the
a
left of the
screen.
slider.
Where is your mouse?
I'm moving my mouse, but I see some pictures in the middle. So I mean pictures of us in the middle.
In the
No,
bottom
we
of the PowerPoint, there is a slider. Do you see the slider at the bottom?
We do not see that, Mark.
Okay. Can you see it now?
Okay, right here, to the left of the slider, yes. Bottom.
I see your pictures there. I don't bunch.
Yeah, to the right, just a little bit to the right. Click that button where there is a screen on the tripod. To the right of the book, between the slider and the book. You can move the pictures away.
Oh, I can remove them.
Yeah.
How do you remove them?
Okay. All right, that's I guess okay. You can just
good
go
enough.
like that.
Yeah.
Oh wait, on the top of the screen there is also like save button, third button on the right after the save button. You see the save on the top?
No.
On the top there is a save button. On the top, pink purple button on the top. You see it? Autosave. No, left. Pink, purple on the left.
Left.
Save. And to the right of the save there is a show button.
Oh, okay.
No,
this is something else. Sorry.
All right, let's go back. Yeah. have two screens apparently.
Окей.
Ну хорошо. Закрой comments, это будет нормально. Close the comments, and that would be fine. Comments on the right, just close them, that's it.
Press escape.
Close the comments to the right, there
There
is
we
a
go.
cross
Good.
button.
No. Press escape.
Окей, I press escape.
Okay, good. We can start. We can use that. That's fine. We can see it as long as you can advance the images with your
Okay.
on your.
Yeah. Oh, okay. Okay, I think I'm slowly
Make sure you can
improving.
go on to the next slide before you start.
Yes. So a minimal scientific model of reality
Okay, go for it.
to account for remote viewing and applied mathematician's perspective. Abstract. A minimal conceptual mechanistic model of reality for remote viewing is formulated based on a high-quality typical precognition remote viewing study. The key idea: reality consists of two distinct interactive components: space-time, plane, abbreviation ST, and non-space-time realm, abbreviation NST. The remote viewing information is obtained by the individual's directed consciousness, guiding their non-sensory perception to cross interface between space-time and non-space-time. The model is based on application of the phenomenological reduction following Husserl and Merleau-Ponty. A conceptual operationalization of consciousness is given. Current scientific objections to remote viewing are addressed, and an example is given offering a comparison between the mechanism of remote viewing and that of scientific discoveries. So my talk would be very different from what you guys have been doing here. Okay, so motivation. If we consider this problem, which is mentioned in the abstract or remote viewing, so we consider from two perspectives. First is from epistemological. From the epistemological perspective, I quote Wittgenstein. It was written about 100 years ago. We feel that even if all possible scientific questions be answered, the problems of life have still not been touched at all. So this is our epistemological problem. So what is our approach? Well, our approach is the following. We borrow the ontological problem from Maharishi Mahesh Yogi, which I discussed. And in our analysis, we use essentially approach of analytical philosophy. That's why I said applied mathematics approach. And we use some phenomenological reasoning. So first, the quotes from Maharishi Mahesh Yogi, which will be the basis of our model. So being lies at the basis of everything, beyond all relative existence, beyond all forms and phenomena, because it has its pure and full status in the transcendent. It lies out of the realm of time, space, and causation, and out of the boundaries of the ever-changing phenomenal field of creation. Existence, life, or being is the unmanifested reality of all that exists. It is the source of all time, space, and causation. So this is the foundation of our ontological perspective. And one more quote of Maharishi, this is just to inspire me for this work. The abstract concepts of mathematics have their life at the source of the laws of nature, perfect models to predict the behavior of everything in nature. Okay, so now basic question to you guys: is consciousness primary? Some people believe it is. So, and my answer as an applied mathematician is, depending on what is. And this is probably a familiar for all Americans sentence, right? Depending what is. Okay, so formulation of the problem. So first, what is remote viewing in this context? Remote viewing is the ability to perceive information from a distant location without using known sensory channels. In remote viewing state, mind is turned off. That is, the conscious mind's analytical and judgmental processes are minimized to allow the subconscious impressions to surface. Some research indicates that successful remote viewing may involve default mode network activation. This is typically known from in some meditation, this mechanism works. Okay, so let's look at specific examples. So our model is to account for specific example. So this is our aim. Since I'm an applied mathematician, so I take an example. What is minimal mathematical model to explain this, to account for this example? So remote viewing example, a precognitive remote viewing. For those who are not familiar, the word precognitive means view something which has not happened yet, and view it in, view and predict what it will be. So here is a short abbreviated description of this example. A viewer is asked to describe a locale depicted in a picture which they will be shown to them in an hour. So they, in simple English, so they are asked to describe the picture which they have not seen it, but it will be shown to them in an hour. Now, in this picture, let's look how this picture is selected. It's very interesting. So this is an outline of the remote viewing protocol. The viewer made a simple, so first what the viewer did. The viewer made a simple drawing and an oral description. After the data has been collected, watch what was done. A computer program automatically selects a pool of 72 targets and a 5-photograph target set. A judge, blind to all that has gone before, compares the session data to each of the photos using a formal procedure. At the time previously agreed, a random number generator selects which local photo is the target. And it is displayed as feedback for viewer. And the result is, description was very accurate, detailed, and judge made first place, which means high quality. Is the idea of this experiment clear or not? Okay, no comments, so I assume it's clear. So how to explain it? Okay, formulation of the problem. Okay, we start with what are current scientific objections to remote viewing. Remote viewing, it has subjective nature, lack of testable theory to explain the phenomenon, and reproducibility, the difficulty to replicate results. Okay, very reasonable. Our aims: formulating a minimal conceptual mechanistic model to account for the remote viewing example. Show how our model can be the first proof of concept step for developing a general model of reality. This is something I will mention only very briefly at the end, but focus, of course, on the first part. Okay. Okay, so the method I'm using to develop my model is very straightforward, but it's may not be very well known. It's a phenomenological approach following Husserl and Merleau-Ponty. So I briefly summarize here some information which some fact about phenomenology which I'm using. Okay, so now why I'm doing it? Well, on the one hand, our model is very short and logically straightforward, as you will see. But on the other hand, the model shatters our natural attitude and implies a radical change in our sense-making of reality at the deepest levels. And to place our model into the proper perspective, we summarize below some basic phenomenological background which we use. So what is phenomenology? Phenomenology is concerned with describing the structures of experience as they are given, without interference from outside theories or metaphysical assumptions. And we summarize some statement from Husserl and Merleau-Ponty. So first, natural attitude. Natural attitude, this is all what we all have. Actually, all the science is natural attitude, is using natural attitude. This is the philosophical stance where we live in the world without questioning its existence or the validity of our perceptions. We take things as they appear and act accordingly without pausing to analyze the conditions of our experience. Consciousness is just a process of registering and interpreting the world without pausing to reflect on the nature of experience or perception. Husserl phenomenological reduction, what is it? Consists of two parts. The first part, transcendental attitude, which has a name named Pekker. It involves a deliberate suspension or bracketing of the natural tendency to take the world and its structures for granted, as it is in the natural attitudes assumptions. Transcendental reduction is a reflective process of questioning the origins of our experience and the way we perceive the world. It is a move towards understanding the conditions that make our experience possible. And next step is eidetic reduction. We focus on the essential structures of experience itself, aiming to understand the core, invariant features of the phenomenon, distinguishing it from the accidental aspects. By suspending the natural attitude, awareness of the reflective structures that, wait a second, sorry, I missed one slide. Okay. Now, Merleau-Ponty approach, it's a little bit different than so, okay. Merleau-Ponty's approach is inversion of the natural movement of consciousness. And he means by this something very simple. This involves intentionally shifting attention from the object of consciousness to the act of consciousness itself, to the way the world is experienced that allows for breaking free from the habitual way of perceiving the world and the self, to reveal the foundational structures of lived experience. The aim is to make the structures of the world and experience accessible for description. The inversion allows for stepping back and examining the foundations of experience to investigate how the world appears and how people are in the world. By suspending the natural attitude, awareness of the pre-reflective structures that underlie perception and interaction with the world can be gained. This includes recognizing embodied being in the world, the fundamental connection between bodies and experience, which traditional philosophies often overlooked. The inversion leads to the discovery of radical situatedness within a world that precedes efforts at understanding, but to which people are also fundamentally bounded. Now, Husserl concept of objectivity is not about a mind-independent reality, but rather an intersubjective one, constituted through the shared experiences and intentionality of conscious subjects. It involves a process of bracketing or suspending our natural attitudes and presuppositions about the world to examine the structures of consciousness that give rise to our experience of objects. Objectivity is not a pre-existing given, but rather a product of consciousness and intersubjective experience. It is through the shared structures of meaning-making that we come to understand and experience a world that is both objective and intersubjectively valid. So to summarize, Husserl's concept of objectivity avoids both the pitfalls of pure subjectivism, where reality is solely dependent on individual consciousness, and objectivism, where reality is entirely independent of consciousness. Understanding of subjectivity in the context of scientific discoveries. So I will have at the end an example comparing remote viewing with scientific discoveries, and this is what I will be using from Husserl. Subjectivity of the ground is the ground of science. Scientific objectivity itself is constituted by and grounded in transcendental subjectivity or intersubjectivity and intersubjectivity. All scientific methods and experiments are inextricably linked to the consciousness of the observer and the operations taking place. Therefore, science ultimately finds its basis in human subjectivity, but in a transcendental sense. This is the critical word. In the transcendental sense, as you will see, distinct from mere individual psychological states. So this is the most important point, and you will see how it's used. Intentionality and objectivity. Consciousness is always directed toward an object. Scientific judgments, despite aiming for objectivity, originate from subjective sources involving meaning-making and meaning-fulfilling intentional acts. To truly understand how objective scientific judgments are formed, one must investigate their subjective sources. Okay, so we are done with all the preliminary. So now derivation of the model. So, okay, an applied mathematician's approach. What does it mean? Okay, what do we mean by this? It means look at the data and uncover a minimal assumptions, mechanistic model that accounts for this data. Let's define all the terms. What do I mean by mechanistic model? Mechanistic model is defined as one based on cause and effect and classical logic. And the method used is to uncover. Here's the word uncover, yeah? What do I mean by uncover? Uncover in the sense of phenomenological reduction, Husserl and Merleau-Ponty. Okay, so we have empirical data. What is the empirical data? A viewer has accurately described the locale depicted in a picture, which will be shown to them in an hour. Is this clear for everybody? Just, I want to be sure that I did not lose anybody. Is this sentence clear for everybody? Okay. So derivation of the model. A minimal conceptual mechanistic model of reality for remote viewing. Ontological assumptions. Reality consists of two distinct, disjoint interactive components: space-time plane, abbreviation ST, and non-space-time realm, abbreviation NST. We call interface the boundary between space-time and non-space-time. So what do we know about non-space-time? We know that its content includes information about past, present, and future in space-time. So what was, is, and will be in the space-time. Epistemological assumptions. Our consciousness in space-time directed towards an intentional object in space-time can cross interface via our non-sensory perception and access, which means perceive or become aware of some content in non-space-time. The above content from non-space-time is then actually constituted by our consciousness in space-time through our experiences and intentional acts into a space-time content and expressed and communicated in some form of human language using one of our or some of our 5 human senses. Crossing of interface cannot be controlled by any actions in space-time, though some actions in space-time may facilitate this crossing. I remind you, it is interface between space-time and non-space-time. Definition, which is a conceptual operationalization of consciousness. Consciousness is defined as a human faculty, the capacity for awareness, subjective experience, goal adjudication, and information processing. This includes using senses, our senses in space-time, and using a non-sensory perception, which is a faculty of consciousness, for crossing, either being aware of it or not, the interface. Discussion. So this was the model. It was a conceptual model. Example 1, comparison of remote viewing example and 2 examples of great scientific discoveries. Okay, remote viewing example. Description. A viewer has accurately described a locale depicted in a picture, which will be shown to them in an hour. Two examples of great scientific discoveries. For both of them, it was self-evident that their discoveries came from a higher source. Okay, first is Isaac Newton. And he was, as we know, deeply religious with complex unorthodox beliefs. He discovered the law of universal gravitation, which was a mathematical formula, by observing an apple fall from a tree in summer 1666. Dmitry Mendeleev. Dmitry Mendeleev. An Orthodox Christian, he discovered the periodic table of elements in a moment of insight described as similar to a dream in 1869. Okay, analysis. So let's see what's similar, what's different between those and remote viewing. Similarity. In all three cases, the information did not exist in space-time, and hence it could only be accessed from non-space-time via a non-sensory perception by consciousness directed towards its intentional object. So this, in all three cases, the fundamental mechanism is the same. So what are the differences? Here are the differences. In the case of the remote viewing example, intentional object was consciously crossing interface via a non-sensory perception to find a particular information in non-space-time and report it back to space-time. So this is remote viewing. Now, in case of bioscientists, intentional object was uncovering foundational structures, the core invariant features of the phenomenon they were studying in space-time. So basically, what were their scientific activities? They were focused totally on their scientific research. In the process of their intellectual contemplations, both unconsciously crossed interface. So in all the cases, everybody accessed the information from the non-space-time. So in the first case, consciously, in the case of scientists, unconsciously. The mechanism is, on the fundamental level, is the same. Okay, example 2. Current scientific objections to remote viewing and our response to them. Objection 1, the subjective nature of remote viewing. And response to this, a quote from Husserl, because this is based on misunderstanding of the nature of how science works, which is currently basically in all our science. Objectivity is not a pre-existing given, but rather a product of consciousness and intersubjective experience. It is through the shared structures of meaning-making that we come to understand and experience a world that is both objective and intersubjectively valid. Husserl's concept of objectivity avoids both pitfalls of pure subjectivism, where reality is solely dependent on individual's consciousness, and objectivism, where reality is entirely independent of consciousness. So this is the answer. Just quote from Husserl. Objection 2, the lack of a testable theory to explain the phenomenon. A testable theory is formulated was just formulated, actually, the outline of the theory. It explains the phenomenon, but it provides explanation, though it is an outline, of course. Objection number 3, reproducibility. The difficulty in replicating response. The key components of an remote viewing protocol is crossing interface, which cannot be controlled by any actions in space-time, though some actions in space-time may facilitate the crossing. This has been carefully considered when designing research protocols. And the last example, just to have some fun, the most famous case of phenomenological reduction. It can be interpreted as eidetic reduction according to Husserl, or inversion of natural movement of consciousness by Merleau-Ponty. And the essence of it, unconsciously crossing interface. And everybody will recognize what it is. We hold these truths to be self-evident, that all men are created equal, and they are endowed by their creator with certain inalienable rights, that among these are life, liberty, and the pursuit of happiness. And finally, remark to finish my talk, a natural extension of our model would be to add additional ontological assumption, namely that space-time, reality in which we live, is a process of continuous materialization from non-space-time. If we accept this basic assumption, then as a corollary, we get trivial explanation for all psi phenomena. And thank you for your attention, and thank you very much for inviting me to make this presentation. I'm ready for your questions.
Well, thank you. Excellent. Mark, that was exactly 30 minutes. And we have 10 minutes for discussion. So the floor is open for discussion.
Glenn, may I ask a question?
Of course.
That's Vladimir. Yeah. Do you hear me?
Yeah, yes,
good.
Yeah.
Yeah, Mark, thank you very much for your very, very interesting and inspiring presentation. My question is, what is the difference, or there is no difference, between telepathy, which we have to believe now, not believe, but to live with now, and remote vision?
Well, yeah, there is difference. In remote viewing, you have specific intention to obtain information about some object, either it's present or in past, on future. So this is the intention is. And telepathy is sort of more general term. It can be a special case of telepathy, for example. But telepathy seems to be more general to me.
It is more general.
Yeah, because it can include anything, sort of more, I would say more generic, maybe, yeah.
But do you think it's the same mechanism that's involved whereby consciousness has to physically leave one body and go to another place?
Yes, exactly. And I want to add one more thing. There is no transmission in this. This is a very important point. It's only materialization. There's no transmission from one point in space-time to another point in space-time. No, it's a process of materialization from non-space-time into space-time. And of course, there's a feedback, but this is beyond this talk.
I agree with that.
So, Mark, do you think that there's any kind of intervention, whether chemical or energetic, that can facilitate this process?
Well, yeah, of course. I mean, we know it also depends on people's natural abilities. Yeah, different people have different abilities and different maybe training. So yeah, I think all of us are familiar with some of those methods, and we know that there's some other methods.
Okay. Are there any other questions? Process.
Yeah, may I just add to the final remark? We were talking a lot about that, about some phenomena, and we were considering them, these phenomena, as general. You see, from my point of view, there is one mistake here. People are very different. For example, when we were talking about consciousness and how to affect consciousness and so on, you see, we have 8 billion people on Earth. They're very, very different. Of course,
yeah, I agree 100%.
Yeah, and some of them, some consciousness of some people is not higher than consciousness of worms. You see, but we should pay attention to those whose consciousness is human. Marx was talking about that we are separated and so on. A lot of people are separated, but a lot of people are working in concert, in orchestrated. So we should differentiate. That is, from point of biology, that is very important. When we unify everything, we made a great mistake. The strength of development, we call it evolution. In fact, evolution is development. It depends on those who deviate strongly from the average.
Well, I would say it depends strongly at quality of their connection with higher realms, which are outside of space-time.
Yes, exactly. When I'm talking about differentiation, this differentiation is in many different aspects. And one aspect is what you were talking about. For example, I wanted to ask you, do you, of course, you know the Messing, Wolf Messing.
Yeah.
For sure, he was exception, one of the very rare exceptions of those with remote vision.
Yeah.
But there are not very many Messings among us. But he is an example of what human being may be like.
Yeah, but this is, I think, yes, this is true. And I would say that this is not really important for us. This sort of can be a distraction, because important is our connection with higher reality, which is beyond this. And in this sense, each of us has this potential, and some people develop more than others.
Yeah, that's what I say. Yes, I agree with you. I think that Wolf Messing was one of a few who were who didn't have boundary between these reality, our ordinary reality, and well, you another term of non-reality. There was no boundary for him personally.
Yeah.
Let me comment on the role of DNA and tools to achieve special states and remote viewing. So DNA is a portal in a way that it connects macro scale with microscale. The width of DNA is 2 nanometers, which is pretty much a quantum scale where certainty is impossible. It's a probabilistic wave function is very diffuse. So there is pretty much nothing sitting in place. It's a probability wave function defined where atoms and parts of the atoms are. And the length of the chromosome is about 10 centimeters, which is very macro scale. So DNA is a transmitting substance which transmit quantum events to macro events and backward. Other things do that as well, like water has macrostructures and microstructures, and also is a combination of quantum and classical physical phenomena, and pretty much the whole biological body is a transmission tool. But DNA is special because it has a very perfect structure with a code, and everything else is not as perfectly coded. Like every cell has about the same sequence of DNA, while the other things are not as perfect. So that's one of the things. And second is that the special states require loss of ego and loss of focus, either extreme focus or loss of focus. It's like two ways to achieve the special state. And that happens in a dream for sure. But in a dream, it's really hard to bring it back from the dream. So we're alternating between the dream state where everything is delocalized and the whole brain and body goes into a special state where things connect together into a united vibrational network. And then awakened state where things are disconnected and there is a lot of certainty. And our culture demands certainty. The civilized life demands certainty. So the trick to go into a special state is allow for uncertainty, which for many is painful and it's equivalent to sickness. But that's what is trained in special spiritual arts such as arts of meditation and yoga. So there you allow for loss of self and lots of uncertainty. And that uncertainty, I think, is like a collapse of function, which is even visible in the structure of the brain and the structure of DNA. So oscillating between dream state and awakened state is a trick for telepathy, remote viewing, and other psychic arts. And Wolf Mason was trained to go in a special state in a circus. So he was demonstrating this samadhi state, a special state of sleep. And that training in the youth, in the childhood, helped him to do it perfectly in adulthood. It was his mechanism of survival. He had to do it. And in this way, I put everybody to sleep.
Yes. Is it time for you already? For you, it's too early, I suppose.
We
for
have
us,
11:30 in the morning, so it's
I see.
midday.
Yeah.
But I woke up around 4 a.m., so I'm a bit spaced out.
Okay, we have 9:30 p.m.
So we have a few more people with the cameras off. Andrew, Irene, Lucette, Mark, Anna. Hey, I can see you.
Привет. Мне все очень понравилось.
Do you want to do any comments before we close?
Ну,
Pick up.
yeah, because it's mostly like Russian speaking here, right?
It's still will go into recording, so I would suggest staying in English.
So I just was wondering if we can kind of combine and collaborate all our efforts and create some portal, like on whenever Telegram or whatever it is. And
yes, people suggested Telegram group. Yeah, we can do that.
yes,
I think I already created, and I will send an invitation. I think maybe I will send it again.
Oh, cool. Yeah, and we can do experiment, in the lab or something. Yeah, like if you have guys idea, then I have I can one sec. So I can go to the lab, and you also probably, Glen, because we have different facility and different equipment. I'm here. I work in university, so this is the lab. So it's a lot of different equipment, which is just, so maybe I can use this. Yeah,
Sounds
just
attractive.
yeah, come
have
come
ideas.
over. Yes, I invite you, and this is for cells and cell incubator.
Yeah, one
Yeah.
of the basic experiments I wanted to do is to do a PCR where you allow different, you connect optically different PCR reactions and see if they can talk to each other.
Oh, wow.
And another one was measuring quantum entanglement in cell culture. It's pretty easy, actually. You first quantum entangle cell culture, and then you separate them and see if quantum entangled couples are talking to each other while not entangled don't.
Oh, did you already do that?
No, these are ideas. I need hands and somebody
I
to
would
do.
love to. I would love to.
Sounds great.
This is actually, I was thinking, what can we do? And yeah.
Yeah, these are very cheap basic experiments. Just grow cell culture, quantum entangle them by just having them contact each other and then separate. And you have controls which won't quantum entangled. And you have experiment where two cultures are quantum entangled, and you treat one of them and measure the effect on another.
Oh, this part I did already. I did it like 2 years ago in a different lab when I worked for the other company, and it didn't work.
There was a series of works of one Switzerland. I just forgot his name. I knew him very well. He was working with this quantum entanglement, and his papers were published in PLOS ONE. When I have his works in my database, I don't remember what was his object, probably some amoeba or other animals like this. So he published it, and he had seen this quantum entanglement. And in general, this quantum entanglement is known from general biology. This is so-called group effect. The group effect, yes, that living systems behave differently depending on the quantity of individuals in a set. Even if these individuals, like for example, if you take one egg of triton egg, it will vary. There is a problem with its division. If you take 2, 3, 4 eggs, they divide much easier. If you take, if you increase the quantity above some threshold, there is again a problem. And even this happens. This is a group effect, and it happens even if they are separated. They live in two different vessels, but not so far different from each other. There is the phenomena of group effect is very well known, but by very few biologists, as a matter of fact. Though it was discovered by Remy Chauvin in 1930s. This is a French, great French biologist who has shown that behavior of population depends very much upon the quantity of individuals in this population, and there is self-regulation of the multiplication. So this is completely against all these Malthusian ideas. And it was there is optimal quantity of individuals in the population for its survival for a long time. The multiplication depends on the quantity of individuals and not on the quantity of resources. It has nothing to, of course, if there are no resources, it will die out.
Oh.
But in principle, if there is excess of resources, it doesn't affect the quantity of population. So the idea that the quantity of resources is just some limit for the development, it is a wrong idea. Though we believe it's still, we were taught that we depend very much on material resources, energetic resources, and so on. This is completely wrong. We depend upon our interaction with each other.
Wow. I never heard of this. It's just like,
Of course, nobody has heard about this because this neo-Darwinism rejects, not Darwinism, but neo-Darwinism rejects it completely because of the political and emotional and so it's a common sense ideas. It has nothing to do with biology. It has to do with our education.
I think it also has to do with our social implants, means the
Yes, yes, yes.
program has been implanted since childhood. That's what I was talking in, and Max was talking like about 2 hours ago about the theory of universe, right? Everything. So, and it depends on if we can rewrite these programs. And I guess
it is very different. No. Well, again, your idea to rewrite these programs, I think it is, well, it cannot work in principle because we all depend on the general field which we cannot govern. We can only study it and behave according to this field. Usually, I mean, about which Sergey was talking a lot, this biological field and so on. We should understand that this is biological field, which is the not paradigm, but well, kind of a route according which we should go. If we go opposite to it, then we have problems. We should understand this biological field and try to behave according to it. We should behave according to nature, which we don't know. The thing is that we very often behave according to our fantasies, which we believe that due to our rationality, we can devise everything.
Yeah. Right.
Totally agree.
I wouldn't call it fantasies. I would call it
they're fantasies, in fact. If we don't know nature, then we construct it. And our constructs are fantasies. And we begin to behave according to our fantasies. And that is our problem. That's why we have so many problems.
We should all go to India and meditate on
not only. Probably, but it's different culture. We should use science in order to understand how what are real natural, not laws, but as Sheldrake says, intentions.
Intentions, right?
Habits of nature. That's what I believe in.
You know, just some crazy thoughts sometimes come to my mind, and I was thinking that all these rules, for example, the golden ratio and all equations and phi and the whatever he showed to us, would it work on the line of horizon, for example, because they don't have the concept of time. So it's only in our universe.
Well, first we should confine to our local surrounding. When we're talking about time, each of us has his individual time. In fact,
correct. Yes.
Each community has its own individual time. You see, so time, there is of course astronomical time, which doesn't depend on us, but biological time depends on our activity. And we live in biological time.
Right. I need to
So
process it and look, you know, just listen one more time everything, and then just merge with, dive in it, and
So the
understand
main
it better, because
alternative is either to research or meditate. What do you prefer? Vladimir suggests that research is more
well, both,
yes.
both. You see, yes, there is research and meditation, which we call intuition.
Right.
Without intuition, there is no explanation for intuition. Yes, there is no rational explanation for intuition. I mean, not rational, but better to say the mechanistic explanation for intuition. Intuition doesn't follow from logic. Intuition doesn't follow from all known facts.
Right.
But we need to have both.
I agree.
Without facts, intuition can lead us to, I don't know, to some chaotic situation. But only facts will lead us to stagnation.
And also, it's very important to be surrounded by like-minded people, because if not,
yes, of course.
then
This is a necessary condition, because human being is a social. Well, I wouldn't say the social subject. I would say use the word animal. It is a social subject, not object, but subject.
Subject, right.
Yeah, because without socialization, we disappear. Individual disappear.
Some of us disappear.
Practically all of us. Practically all of us disappear. You see, it's again for the law of biology. There is no individual in biology. That is abstract notion. That is, again, we separate some subject from all surrounding, and we begin to study it. And when we separate, it dies out.
I would say it dries out, yes. So we have to research.
Yeah. Again, and in fact, yes, thanks to Max, and that he managed to to collect people who are similar but very different.
Yes, I admire him and listen to his lectures. He's my guru.
Probably
one, we are united.
Yeah.
It's all illusion. Separation is an illusion.
Yes, exactly.
So the main question is, where the money? Where are the money? Where is the funding?
You know, something never changed. Huh? The same question all centuries, all the time.
How can you meditate to get money?
I don't know.
No, Max, there is no problem in resources. All resources are in our brain.
Yeah, so if money is an illusion, why don't we create such lots of that kind of illusion?
I already started to create it like 2 days ago. I bought the lottery ticket.
Yeah, man. Yeah, investments on silver futures. Edgar Casey tried to do that and he failed. He was the best one, but somehow that you can't really use it, and he wasn't allowed to use it. He could do it for healing of others, but not for financial gain.
Yes, that's the usual situation for people like him, for talented, you know, one of a kind, you know, because if the universe would give him money, like millions of dollars, hundreds millions, then he would lose probably I don't know what would happen. It's my speculation, but probably his passion would cool off. Who knows? Because money is great. It just, when you have it, you got a lot of other interest, and you deviate your attention and scattered and put yourself very
How about
thin.
funding for resonance research?
How much do we need?
A couple million.
Only?
Okay.
I mean,
Well-
after that, we probably would have produced some devices for 2 million is enough to get some patents and get investments. We need a
I
couple
would
million
love
starting
it. If
capital.
look, if I win the lottery and it's 33 million now, definitely the first person I call is you.
Sounds
Should
good.
he work better if you pay him 33 million?
33
see,
million,
I-
yeah.
Yeah, I can tell you, if I get $10 million, should I go and hire Gastarbeiters? I can hire a lot of Gastarbeiters for this money. Should we make science? I doubt.
Yeah.
In order
There's
to
a lot
make
of-
science, you need to grow up your students. And it's not just money, it's brain in the first place.
We have a lot of brain
See,
in university.
great science, as a matter of fact. We very often forget history of science. Great science was made in Russia after the revolution, where people had nothing to eat. I'm talking about great scientists who worked in 1918, 1990, 1920s, where there was nothing practically to eat, but they made great achievements. For example, the Semyonov, the Berg. Well, I can name a lot of great scientists who were working at this time without any funding, or practically even starving. Oh,
So
we don't want to know this, because we think if we cannot make a good job because we cannot buy a device which costs $1,000 million. If I buy this device, then I'll make a great science. I have an experience. I was working for a long time in the Institute of Bioorganic Chemistry of academician Ovchinnikov. It was the richest, probably its resources were the sum of all resources of biological institutes of the Soviet Union. What discoveries did they make besides the primary structure of proteins? Nothing. It was not science, it was sports.
I never thought of this. Yeah.
Yeah, try to think of this.
My best hope is to find investors for some good devices, therapeutic devices.
What kind of devices?
Bioimpedance diagnostic and therapeutic biofeedback, especially the ones which could use DNA resonance. Using quantum entanglement, quantum sensors-based devices, and some sort of feedback that would improve the coherence.
So I guess we have to do venture competition and invite some venture.
Once you have a device that works, it's much easier. So I'm looking for already people who develop them and looking for investors. And hopefully I can merge somebody developed technology that needs more development and then an investor.
Okay, we can hope for a long time, but
Yeah,
probably
we can
it's
pray.
already, yes,
can
we can pray, but it's already 10 o'clock here in Moscow.
It's
Vladimir,
3
thank you very
o'clock
much.
in
But if you
Miami.
know someone who has technology that and is looking for investors, I would be happy to meet with them and try to facilitate that.
All
Now I will
technologies, tremendous quantity of money is for prolongation of life of a human being to infinity. Probably you've heard about this project.
No,
Of a particular human life to infinity. So the tremendous quantity of money goes into this program. So I stopped talking on this topic. That's all. Don't ask questions. I think you can devise what I was talking about. And
Okay.
fortunately, I believe that biology has a ban on such projects. If you have even billion of dollars or trillion of dollars, biology is a ban.
And
Okay.
with that, we
Let's just
close
put
Thank you very
the
much.
session. Thank you very
Yes.
much, everybody who is still with us. And
Yes.
press the button. You know how to find us. It's dnaresonance.org. And please subscribe to my mailing list. So send me an email to max@dnaresonance.org. Have a good day, everybody. And if you have
Спасибо.
with
Thank
us,
you
let
so
me
much.
read the list.
Thank you
Thank you,
very much.
everyone.
Just let
Thank
me
you very much.
see.
Thank you.
Get Anna, Mark, Vladimir, Andrew, and Lucette.
Yeah,
Goodbye.
bye-bye.
Thank you very much.
Goodbye. Bye-bye.
Thank you. Vladimir, were you